Καλησπέρα σας. Σας καλωσορίζουμε στην πρώτη Cannabis Excellence Day της Hellamco και σας ευχαριστούμε για τη συμμετοχή σας. Hello, Zdravo. Welcome, Dobre doje. Welcome to the first Cannabis Excellence Day organized by Hellamco, and thank you all for your participation. Although we have met with most of you, let me introduce you. My name is Christos Kondogiannis. I am the General and Quality Assurance Manager of Hellamco. During our vast 38 years of experience, we collaborate with internationally recognized manufacturers to supply high-tech instruments following their strict specifications and standards. The company provides solutions both to quality control and to research and development laboratories, with complete proposals tailored to the needs for each customer, and so we enjoy a leading position in this market. Following now the modern needs, Hellamco expanded since before 2010 in the scientific field of cannabis and has successfully installed some instruments. Specifically, Hellamco is the only Greek company that provides all the necessary equipment for the cultivation, the processing, quality control of integrated pharmaceutical and industrial cannabis installations. We support the efforts of the Greek, Cypriot, and North Macedonia scientific community to expand the research for developing new medicines and products to improve the effectiveness of final products and ultimately the human health. Based on our significant experience in organizing scientific seminars, webinars, training sessions, we wish to transfer you the latest developments in the field of medical cannabis and hemp by some experts in this field. Our first Cannabis Excellence Day is organized with the participation of very important manufacturers with whom we have exclusive cooperations. The 9 speeches are presented by 11 specialized executives from Europe, Canada, and the United States. SEM Corporation, CONVIRON, MOBIUS, THAR PROCESS, VALOYA, VTA, and Waters are some of them mentioned today. We tried to cover as many topics as possible and made sure that each presentation is as short and concise as possible, while any question you may have at the end of each presentation will be very welcome and treated as confidential. Due to the limited time, any answers that are not covered at the end of each speech will be answered later on and sent to everyone. We believe that the whole effort is very interesting and useful for your work, and we will sincerely appreciate any suggestions for improving the next cannabis seminar, hoping that it will be established on an annual basis. For this purpose and for your convenience, a relevant questionnaire will be available at the end of the session. For those participants who wish to receive a certificate of attendance, it will be issued upon completion of the seminar, and there will be also a recorded version for those who cannot connect live today. So really thank you once again. We're really excited for this today's seminar, and let's now move on to the first speech of the day. Our colleague, Mr. Yannis Mouroutis Prionistis, chemist, sales manager of Pharmaceutical Cannabis, will present you our speakers and will help in submitting your questions and organize the answers if there is left some time at the end. So thank you once again. Lagodaram, please, Yannis, continue. Thank you.
Thank you very much, Christos. Good evening also from my side. Most of you know me. My name is Yannis Mouroutis Prionistis. I'm a chemist and I'm sales manager in Hellamco North Greece office. Besides that, I'm a business unit manager for the pharmaceutical cannabis section of our company. I'm also excited for this first Cannabis Excellence Day that takes place in Greece. It's the first similar webinar that is happening in the Greek market, the Greek cannabis market. And we don't know if there is something similar that has run in the European market. The purpose of this seminar, as Christos told you, is first of all to provide you our solutions in the cannabis industry through our partners and help you design, run, and keep updated every pharmaceutical cannabis industry you're going to obtain. So let's start with the first presentation. Axel Sonner will be our first presenter. Axel Sonner is the Director of Distributors Sales in EMEA and Southern Asia from company SEM. And his presentation will focus on sample preparation for detection of pesticides, mycotoxins, cannabinoids, and heavy metals in the cannabis industry. Axel, you can continue.
Yes, thank you very much for the introduction. Good morning, everybody. Good afternoon, everybody. I'm sharing my screen now, hoping that everything works. And I'm going to move to the presentation mode. So can you see my title slide?
Yes, perfectly.
Okay, so the title of my talk today is Rapid Sample Preparation for the Analysis of Cannabinoids, Pesticides, and Heavy Metals in Cannabis. Basically, CEM is a US company and specialized, among other techniques, in rapid sample preparation, which is the focus of today's talk. I want to talk about two methods for different parameter testing on cannabis. We will look at solvent extraction techniques for the determination of potency, pesticides, metals, mycotoxins, terpenes, solvents, and microbiological screening. For these parameters, potency, pesticides, metals, and mycotoxins, and terpenes, we do have two technologies available. One is accelerated and pressurized solvent extraction, and the other is microwave digestion. I want to quickly show these two machines here on the next slide. So on the left side, we see the EDGE system. EDGE is an accelerated solvent extraction system that prepares samples for subsequent analysis by GC or HPLC. To the right side, we have our MAR6 microwave digestion system. Microwave digestion is a technique that has been established for more than 30 years as an international standard, and is basically the golden standard for preparing samples to the subsequent analysis by atomic spectroscopy techniques such as ICP, ICP-MS, or atomic absorption. For most analytical techniques, sample preparation is pretty much the bottleneck, which means we lose most time on sample preparation. So when we look at this pie chart here, this is a publication that has appeared quite a number of years ago already in LCGC magazine. The analysis resulted in a part of more than 60% that is, I would call, lost due to sample preparation, whereas the subsequent steps such as data management or the analysis itself and sample collection is less than 40%. So the logical conclusion out of that is to reduce sample preparation time, and we can significantly improve the workflow in the laboratory. Specifically for our new EDGE system, we have developed a couple of key techniques, and one of that is the sample container, respectively the sample holder. So on the left, you see the disassembled state of the sample holder called Q-cup. So the Q-cup is a metal tube with a frit at the bottom, and we use disposable filters to filter the sample at the end of an extraction. So the first step is to assemble the Q-cup, which means we have these two parts plus this Q-disc, which we take this bottom, it's a screw connection. So we put this Q-disc in the bottom and we screw up the on the bottom onto the Q-cup top. We weigh the sample and we place this Q-cup with the sample into the instrument. The instrument has a 12-position auto-sampler rack, and after that we receive a clean sample which can either be concentrated or further processed before analysis by GC or HPLC. A few facts about the EDGE system. It's providing temperature-controlled extractions. To give you an idea, we have a temperature range from more or less room temperature up to 200 degrees. Apparently, for most organic contaminants that we are looking for, we will not go up to 200 degrees. For pesticides, for example, we start extracting at 40 degrees, but for other organic compounds, we can go up to about 110, 120. So if we refer to the US EPA 3546 norm for many organic contaminants in the environment, the typical extraction temperatures are in the range of 100 to 115 degrees. Further details about the edge extraction system, it provides in-cell cleanup. So apart from the filter that I have shown in the previous slide, we can put different types of sorbents. We can put, for example, magnesium sulfate for drying a sample, or we can place silicon earth in order to remove fat. Then, of course, the operation is very simple. It's efficient. The system is automated, and we have less matrix effects in the edge system. I have a short movie. I hope it plays without getting stuck. And at a certain point, I will accelerate this so you can see the system from the front. My colleague is assembling the Q cup. She has fitted the filter already. Now she puts a solid sample inside the Q cup, and she places the Q cup into the auto sampler rack. The racks can be swapped, which means I can prepare racks while one rack is operating. The other rack can be fitted with fresh samples. The Q cups are moved by an auto sampler arm into an extraction chamber. And then the extraction chamber closes and is pressurized. And the system is connected with external solvent containers, so we can connect up to 6 different solvents. And these solvents are individually added, so we can extract with a single solvent. We can subsequently extract with different solvents, or we can prepare a mix of solvents. So there is the capacity of mixing up to 6 different solvents in one extraction. Further to that, we have a dispersive effect. So the solvent is moving up and down, which is controlled by pressure. And of course, the chamber is heated to accelerate the extraction reactions. After the extraction is done, we filter the sample. We push the sample through the filter. And there is at the bottom is a cooling coil. So we receive samples that are cold and are already filtered, which means they are clear and can be further processed either by further cleanup or by concentration, or they can also directly be injected in our chromatography systems. In between each step, the system washes itself. So there is one or separate washing steps, which I'm going to show later in more detail. And when you look at the system there, the bottle on the right-hand side is a waste container. So the wash solvent is collected there. And after the extraction, the sample is collected in a glass vial or a falcon tube, and the sample that has been processed is returned into the auto sampler rack. So it's a fully automated process, which results in a clean, perfectly extracted sample. So this would be the residual sample, and the extract, as I mentioned, is collected in a collection vial or a falcon tube. A few things here. This is a schematic of our Q cup, which is again a metal cup, a metal cylinder. And these are the different components here. We have the threaded bottom when you look at the picture on the left, which contains a filter or a combination of different filters. These filters are called Q discs, and we usually work with a sandwich of 3 filters, which is called S1. And we use a sandwich of a C9, a G1, and a C9 filter. C9 is a cellulose filter, G1 is a glass fiber filter, and C9 is another cellulose filter. We can place a sorbent on top. This could be either drying agents such as magnesium sulfate or diatomaceous earth for fat removal. So we can process these things automatically, and we have then samples that are cleaned up already, which saves extra cleaning steps. In that case, we would have extracted 1/4 gram of cryo-milled hemp, which would be our sample in that context here. And we got this screen on top, which is called Q screen. And the Q screen will avoid that the samples start to float on the solvent. So they keep the sample and the solvent in tight contact. Now, this is a method, how it looks like on our screen on the Edge system. So what we do here, we perform the potency extraction with methanol. We add 15 milliliters of methanol from the top, and we heat this to 45 degrees. We hold it for 5 minutes, we rinse it, and we collect the sample, which means the sample would be finished. On that slide, this is the wash method. We wash with 2 different solvents here. First wash is with isopropanol, and the second wash is methanol. And so the extraction chamber is washed after each extraction. I mean, we do like a suite of extractions. So we do 1 sample at a time and immediately do the next sample after the wash step. So we have a fast sequential operation of the system. There are special reactions to separate unwanted side contents, such as lipids or other materials that are removed by winterization. Winterization means simply that the samples are frozen, which basically precipitates unwanted side products such as lipids, for example. These are some comparative results between the edge extraction and the hand method. And we see typically very good agreement between the different extraction techniques. There is, of course, always some smaller deviation depending on the technique or the comparative technique that is used. But edge is typically equal or even superior in its recovery and its extraction efficiency compared to other techniques. Now this is to go to the next item, and this is preparation for the analysis of metals. So before we talked about organic compounds that are extracted for determination by chromatography techniques. Now we do metals analysis by atomic absorption and ICP. And there are, of course, different things to look at. The plant itself may contain heavy metals, but it can also, there can be additions of heavy metals coming from irrigation water, from the soil itself, from added fertilizers and nutritions, from ingredients used for edibles, oils, extracts, and concentrates, delivery containers or delivery devices, and vaporizers. So the problem of cannabis is it's a hyper accumulator. So cannabis can accumulate contaminants, and in that case, it would be heavy metals from different sources, and all of them would show up. And of course, we have to protect the consumer from absorbing these in his body or accumulating these in his body. There is an actual AOAC official method. It is 2021.03, which is heavy metals in a variety of cannabis and cannabis-derived products. And they are determined by ICP, inductively coupled plasma mass spectrometry. And it can be applied to cannabis products, to plant material, to concentrates, to finished products. And the required elements that are listed in AOAC OMA 2021.03 is cadmium, arsenic, lead, and mercury, and optionally tin, barium, chromium, copper, nickel, silver, selenium, and zinc can be determined as well, which is part of the total metal analysis. The machine we are using to digest these samples is the MAR6, and especially for high throughput labs, we have the capacity to digest up to 40 samples per run. So when you look through the instrument window here, you see a rotor, respectively a turntable, that has up to 40 positions. We do have pre-programmed cannabis methods, respectively plant or dried plant material methods. And these are standard applications. I have to say that neither cannabis or other dried plant material is anything exotic. It is a standard application that is done for many years. These digestions are typically performed in HNO3 and nitric acid, or in mixtures of nitric acid with, for example, HCl or hydrogen peroxide. So there are different chemical cocktails available. I just want to show the different steps, which what we call an analytical chain. So when we come from the raw plant on the left, there is typically a sampling process done by quartering, sieving, comminuting, or homogenizing, which means there is a large, of course, we have a large amount of leaves, for example, and of course the amount has to be reduced because what we need for digestion is about 0.5 gram or 1/4 gram of material. So coming from the raw plant or the raw leaves, we have a particle size reduction step, which is typically a cryomill. Why do we use a cryomill? Simply because the low temperature protects heat-sensitive components, such as pesticides, for example. Once we have a fine powder, we weigh it. As I said, 250 to 500 milligrams. Then we add the appropriate acid cocktail, which can be either nitric acid alone or mixtures of nitric and HCl or H2O2. We then place the samples in a microwave digester, and after that, we perform an instrumental analysis, which is done either by ICP, ICP-MS, or atomic absorption. Typically, we have a system that has two important components. We have the microwave oven itself, and we have the vessels. And the vessels are about as important as the microwave oven itself. We typically use what we call mass express vessels. These vessels are extremely easy to use. They consist only of 3 components. So we have the vessel itself, a plug seal, and a screw cover. The plug seal and the screw cover together form a safety valve, which controls the pressure inside. And these vessels are available in different volumes, 10, 20, 55, 75, and 110. So typically for the cannabis digest, we would use 55, 75, or 110, depending on the sample size that is needed. What we normally end up, when we look at the raw material on the left, we need a fine powder. This is how these samples are normally digested, which means, again, we use a cryomill or a cryomilling process. We add the powder into a vessel. We add our acid, typically between 5 and 10 milliliters, and then we assemble the vessel. This is a selection of different materials that we have digested in CEM. And we can actually digest very different products simultaneously. So we have hemp oil, we have actually hemp powder, we have cookies, we have little jelly beans, we have a balm for treating or a pain relieving ointment. So all of these samples are based on hemp. And in a study, we digested all of that in one run. So raw cannabis plus all these processed products together. I would like to show a video here, which I I start a little bit in the middle. This is a vessel, an express vessel, which is disassembled. So we will be adding acid to the sample. This is normally left alone for about 15 minutes for pre-digestion. Then the vessels are placed in the rotor. We have this kind of torque wrench that goes with it. The rotor has, in that case, 40 position. So consider we have 40 samples now. They could be similar or they could be different. The sample is placed, or the samples are placed in the microwave. And then the run is started. We have preloaded methods, which are called one-touch methods that are factory loaded. So we have hemp methods, we have general plant methods. They are very similar. We start this from a touch screen. So method selection and touch screen operation is very simple. And this is normally a process that takes about, I would say, 45 minutes to digest the hemp and other plant materials. So all of that together is quite simple and very convenient. We have, just to conclude this, so we talk about two different techniques: solvent extraction for organic compounds and microwave digestion for metal testing by atomic spectroscopy. A few words about CEM. We are the largest manufacturer of sample preparation systems worldwide. We have worldwide distributions. In our case here today, we worked with our esteemed partner of Hellamco in Greece. We are basically distributed in all major countries worldwide. We do have our own subsidiary offices. Our headquarters is in the US. We are represented by sub-offices in the UK and Ireland, Germany, France, Italy, Japan, and I have to add Singapore, which is not part of that graphic here. As Hellamco is nice enough to collect questions, this is just our generic email address. It's called organic.support@cem.com. We do prefer, though, that you ask your questions through Hellamco so everybody can benefit from the answers. And I am done here with my formal presentation. So I would like to open the discussion for questions. If there are no questions, and this was actually asked in the instructions, I do have a remaining video, which is a complete course on how to prepare samples in the microwave. So if you agree, I'm asking the organizers. The video takes about 12-13 minutes. I don't know, do you want me to play that video? It's quite interesting. It gives a lot of detail about microwave sample preparation and general handling. If not, questions can be asked now or later.
Axel, we can start with the video, and
okay.
I will try to collect any questions. So I will set the questions after the video is playing.
Yeah, fine. So I'm starting the video now. I think, well, I need to share the sound. Oops, you don't hear anything because I don't either. I think I have to unshare real quick to change the sound settings. Okay. Is it to the left?
On the share screen, Axel, on the share screen, in the middle down.
Okay, I currently don't share.
In the middle of the screen, on the bottom, it's a green share screen button.
I need to unshare though because I can't see it.
No? Yes.
So I'm afraid it's not going to work for me here. Okay, I think it works. It should work now. Sorry.
Hello everyone and welcome to our lab. Hello everyone and welcome to our lab.
Can you hear
As
sound?
we were talking about with our AOAC method, you can digest all of these different sample types using our one AOAC method. So some of the samples you might come across are plant material, such as, cannabis plant material. Some high protein samples like cannabis beef jerky. Samples high in carbohydrates like cannabis candy. And samples high in fat, cannabis hemp butter, right? We also have manufacturing intermediates like cannabis concentrates and distillates and isolates. These are some of the more difficult to digest, but we can digest all of them using our method. Some more isolates. And tinctures are also fair game with our method. You have terpene tinctures and CBD tinctures. Personal care products like hemp soap. Also, CBD pain relief cream and lip balm. And last but not least, beverages and food products, so like coffee, wine, any type of beverage you can digest using this method, and any type of food product, cannabis dry food product, you can digest using the same method. All right. We're going to be doing all of this in this Express Plus, Mars Express Plus digestion vessel. All right. It's a 24-place vessel using Teflon 110 ml vessels. They are a 3-piece assembly with a liner, a plug, and a cap. So the liner holds the sample. The plug is for moderating pressure throughout the run, and the cap has a hole in it to allow for venting of gases. Let's say you are a higher throughput cannabis lab and you typically get a lot of the same samples. Let's say you get a lot of plant material samples. You could potentially do this method in our 40-place Mars Express 75 ml vessels, right? These are very similar to the Express Plus. Again, just a 3-piece Teflon vessel. These are only 75 ml, but you can run up to 40 of them at once, and that's big. But you can't just run any 40 samples. You can't run concentrates with your plant material. If you want to run concentrates or oils, you're going to have to run those separately. You can run food, beverages, plant material, personal care products, all of those together in 40 position, but together at once. But you have to run the concentrates and the oil separately. All right? Now let's actually add some sample and show you guys how we do this. So you take your liner, and you will typically, for plant material, we would usually use whey paper, whey boats. You want to make sure that, if you're using these types of things, you want to make sure that you're not running into static issues. So we may sometimes put our vessels in front of an anti-static ionizer like this. That really helps to stop your samples from sticking to the sides of the vessels or sticking to the whey paper itself, right? A good technique for whey paper is to fold it in half. It makes it easier to dispense it into your vessels when the time comes. All right? And there are a few other techniques that we like to use for some of the harder samples to put into your vessels, like the creams or the butters, we typically might use a plastic spatula like this. If you use any type of plastic scupulas or dispenseware, you want to make sure that you clean it beforehand using dilute acid. For liquid samples, we like to use these pipe transfer pipettes. And again, you want to make sure you clean these beforehand using dilute acid, so 10% nitrate will work great. All right, and make sure when you're adding these samples, you get the sample to the very bottom of the vessel. That is very important. You want to make sure that your sample is completely submerged in acid. All right. Some of the concentrates and oils like to stick to the sides, and you want to use these tools to make sure you get it to the bottom. Right. I've got some plant material conveniently already weighed out for me. First let me show you. The plant material, if it comes in a bag, it may not be very homogeneous, right? You want to mix this bag up, tumble it a few times, shake it up a little bit, make sure that everything gets nice and homogeneous in there. Otherwise, your stems might sit on top of everything and the shaky stuff will go to the bottom. You want to make sure you tumble it a few times and get it nice and homogeneous before you sample it. And when you do sample it, don't just take all from one spot. Make sure you take a little bit from here, from there, and get a little bit of everything so that you get a nice representative sample. All right, so we take our pre-weighed out sample and add it to the bottom of the vessel. All right. Let's take this over to the hood and add acid. So, for our method, we call for 9 mils of nitric and 1 mil of HCl. You can see here we have acid in glass bottles. We do not want to use acid in glass bottles. Glass is inherently dirty and it will contaminate your samples. So, we tend to use either our own distilled acid that comes in Teflon bottles, or you can order your own ultra pure, reagent grade or trace metal grade acids from a few different sources. All right. So for adding the acid, we tend to use micro pipettes, just because we know the tips are clean and we know they're very accurate. Let me go ahead and add some acid. Now, once we've added our acid, we'll let it sit and pre-digest for up to 15, maybe even 30 minutes, depending on how reactive your sample is. If it's reacting at room temperature, it's a good idea to just go ahead and let it pre-digest. If it's not, then you can just go ahead and seal it up and go. But for most of these cannabis and cannabis-derived products, they're going to react. So just let them sit for at least 15 minutes. All right. One thing you'll notice here is, actually I have some cannabis plant material already added to this tube with acid in here. This is 10 mils of acid. You can see, actually, that the plant material is creeping up out of the acid, right? That's not good. You want to make sure that it's submerged. So you might swirl it around it a little bit just to get it all mixed together so that it runs well. You see? It's nice and submerged. All right? What we do not want is to use too little acid, because then it will all be spent during the reaction. You can see here, this is only 3 mils of acid. It's not going to completely submerge in 3 mils, and you're going to run into problems, and it's just not going to completely digest. So, to get around that, we recommend using at least 5 mils of acid. We would prefer that you would use 10. All right, so now that we've got our sample pre-digested, let's go ahead and add our plug on top, and then pour down our cap. If you gave this to 5 different lab workers, they would all hand torque it down at 5 different pressures. So what we want to do is use our hand torquing device so that every vessel gets the same amount of torque. And what that'll do is prevent certain vessels from taking off and overreacting while others don't. It'll make sure everything is nice and flat and reacts well together. All right. So now that we've got our vessel torqued, let's go ahead and put it into the turntable. Make sure it's nice and flush, and then we'll take it over to the microwave. All right, so we'll take our turntable, put it in the microwave. Shut the door. Then you have two different method directories to pick from. We're going to pick the one-touch directory. That comes with all of CEM's pre-programmed methods, and you'll find the cannabis one-touch method near the top in the C's, obviously. You press that method, and that's it. You hit start, and the unit will determine how many vessels you got in there, what kind of vessels you're using, and how much power to apply so that you get there safely, right? So let's go ahead and look at the run history of a previous run I've run. You can see here the power starts off low, as does the temperature, which you would expect, and it slowly ramps it up to temperature. We call that ramp to temperature. It's a very safe way to get our samples all to temperature, everything relatively at the same temperature and same pressure safely. So once we get to the end of the ramp, we're at 210 degrees, and you'll notice that the green line, the power there, is modulating to make sure that the red line, the temperature stays steady. All right? We want to hold it at 210 for 15 minutes. After that, the samples have digested. They'll go into a 15-minute cool down, and after the 15 minutes is up, you're able to pull them out and take a look at your digestions. Here I'll show you some of the details of our run previously. We ran 14 vessels, and you can see here the check marks. That just shows that your sample made it a temperature. If you see any X's there, it'll also give you a temperature that vessel reached. And that way you know, oh, my sample did not make it a temperature, that's why it did not digest. Really helpful tool. All right, so now that our samples are digested, let's pull them out of the microwave and take a look. All right, now that the reaction has subsided, there is still pressure in these vessels, right? We want to make sure we're safe when we open them. Again, there's a hole in the cap to allow fumes to vent out. You want to point that away from you and vent in a fume hood, right? Like this. You can see the gases coming out of there. That's what you should expect. There will pretty much always be some type of pressure built up during the reaction. It's usually carbon dioxide and NOx gases that are formed. All right. Now that we've opened it up, we want to dilute this up. And the way you dilute up matters. You don't want to use any type of glass. So the two different ways you can dilute up are volumetrically and gravimetrically. If you're using volumetrics, you want to make sure you use Teflon volumetrics that are pretty clean. Rinse them over a week in dilute acid before you use them. Don't just reuse and reuse because you'll get contamination from your samples. All right? Dilute to the line. This is a Class A volumetric. You want to make sure you use Type 1 ASTM water. Another way to dilute is gravimetrically, and this is what we use in our method. It tends to be more accurate. That's why we use it. You want to make sure you pre-soak these containers and dilute maybe 10%, 5% nitric acid for a day or two before you use them, just to make sure they're clean, even though we're pretty sure the ones we get are clean beforehand. So you'll pour your sample into there, and you want to quantitatively transfer. We typically do 3 rinses out of the liner and into the vessel, and into the centrifuge tube. So obviously you'll have this container pre-tared before you add anything to it, so that you can dilute to weight. For our method, we diluted to 50 mils, using 10 mils of acid. You could get by again though with as little as 5 mils of acid, depending on your samples. All right, so 3 rinses, just to make sure that we transfer everything, nothing's stuck to the sides. You want to make sure that you get everything out of there. All right, and then we'll take it over to the balance and dilute to 50 grams, and we're done. All right, so to recap, you can run all of these different samples. Our method encompasses all the different cannabis and cannabis-derived products, including but not limited to plant material, foods, beverages, consumer products, and manufacturing intermediates, right? So you can do all of these different samples using our Mars Express Plus vessel set. You can also use the Mars Express vessel set if you're okay with taking out your concentrates and your oils and running those separately. All right, If you want more tips and tricks on how to perhaps weigh your samples up, visit our cm.com in the lab for further tips and tricks. And I believe that's it.
Okay, thanks for watching. That was it. Yanis, you collected any questions?
Yes, sorry, thank you very much. We have a couple of questions.
Sure.
Some of them are for Eds and some of them are for the Mars. So I will try to group them. First of all, regarding Eds, asking if we can share more information about results in let's say, compare the results of Eds and other techniques like quenchers or ACE or socket extraction.
Yes, we have such studies available, and I'm glad to share them. This would be by email then, because I would have to look them up.
Okay. So also regarding Mars, we have questions, first of all, regarding safety of the system. For example, what are the safety features we have in order to avoid any pressure or temperature increase?
Well, we have so-called active and passive safety devices. Active safety devices are the control of temperature and or pressure. So this is what the machine does by itself to avoid that we get any critical situation. Second to that, we have passive safety features such as safety valves. So each of our vessels are fitted with a reversible vent reseal mechanism that can release pressure and continue working. We also have safety devices which are built in the system, and this goes a little bit into almost artificial intelligence, because the machine can identify automatically which vessel is being used and how many vessels are used at any time. And therefore, the machine adjusts automatically pressure, temperature, power to the vessel model that it's using. So it has these sensors for auto recognition. Further to that, we have a sensor which is called ReactiGuard. So in case of a strong ventilation of any vessel there, the machine will stop automatically and cool down. So this is ReactiGuard. And we do have also TempGuard, which is looking at all the vessels at any time. And if any vessel is getting much hotter than the others, the system will also stop or run. So there are a lot of different or multi-layered safety features in the machine of the Mars 6.
Also discussing about these features, there is another relevant question. Can the microwave system, the digestion system, control every single vessel individually, or to control the vessels, all the vessels at the same time, regarding pressure and temperature monitoring?
Well, first of all, it depends on the control mechanisms that you have. But the system you have seen just now is permanently controlling the pressure, because each individual vessel is pressure controlled by this special valve mechanism. And secondly, we use either infrared or I-wave temperature control, which monitors every vessel on the turntable. So as the turntable rotates, we have up to 3 different sensors there that monitor the vessels when the vessels are passing over the temperature sensor. So there is a control of all the parameters, which depends a little bit on the control mechanism that is configured with the system, though.
Okay, also another question we have, the last question. Can we simply say that the microwave system is used as a pre-harvest tester, while the solvent extraction is used for testing after harvest material?
Can you repeat the question, please?
Yes. I'm just reading this question. Can we say that the microwaves digestion system is used as a pre-harvest, as a technique for pre-harvest measurements, and the solvent extraction is used for after-harvest measurements?
Well, I wouldn't say that. I mean, you can use any of the system at any, let's say, stage of plant development. Of course, we would assume that the metal content of a plant is going up over the lifetime of the plant. So I would not necessarily measure the plant at the beginning only, because the plant, as long as it stays in the soil, will accumulate heavy metals or other inorganic compounds over its life cycle. So actually, you can measure before the harvest or after harvest, but I mean, it's not limited to that. What I'm saying is, you measure the heavy metal contents most likely when the material has always been harvested, to make sure that the batches are in agreement with the legal limits of the given heavy metals as it's specified in this AOAC norm.
Just to also point something on that, generally heavy metals, pesticides, and mycotoxin detections are used in order to release a batch. So you must have a certificate of analysis and release a batch. So when you are using EDS in order to measure mycotoxins, pesticides, or other contaminants, then you will use it as after harvest. You can use always EDS in order to extract cannabinoids and test also your potency in the sample before harvest, before drying, after drying. In every step, you need to do it.
Yeah, I mean, there's no limitation when you use it.
Exactly. And also, last question that just came in the solvent extraction system in EDS. Do we need to refill the bottle of solvents each time the washing solvent? It is different according, or it is different according to the method.
The bottles are filled when they are empty. I mean, it's as simple as that. You can put large bottles on there, so you have to refill less frequently, or you can put small bottles on there. There is no limitation from us on the size of the bottle you can use. We can connect independently up to 6 different solvents. And typically, for the wash in between, we normally use at least the same solvent we use for extraction, but also preferably a more aggressive solvent in combination with it. But if we use more than one solvent, the last solvent we wash is always the extraction solvent.
Okay. In any case, there are any more questions, you can feel free to send these questions to our email, info@hellamco.gr, and we will forward it to Axel, and he will provide you with the answers.
Okay. Thank you very much.
Thank you very much also, Axel. Thank you very much for the presentation.
Good luck with the rest of your program today. I'm leaving the session now. Thank you very much. Have a good day, everybody.
Thank you very much for everything. It was very informative and very useful to have this information from your side.
Yeah, thank you very much. Bye-bye.
Thank you. Bye-bye. Now we will move to our second presentation. We have 2 presentations from Waters Company. Unfortunately, due to technical problems, these presentations are recorded because Mr. Rainer Turek could not join the webinar. And the first presentation is from, both presentations could be held from specialized people from Waters, focusing on the topics I'm going to present. The first presenter is Miss Emily Rose Britton. She's a senior market manager in Waters USA, and she is responsible for the natural products market in USA. In this case, that we don't have the presenter with us, I will be able to answer your questions, and if they are very specific, I will forward them to Waters and send the answer back to you. So we are going to start our next presentation.
Hi everybody. My name is Emily Britton, and I am a senior marketing manager for foods and natural products within the United States, based in Arizona. And I'm very happy to have the opportunity to share with you today about some of the work that Waters has done to support the cannabis industry, especially related to cannabis quality testing. So what I'm going to present to you today is a application note and solution that was developed by our water scientists in Massachusetts for cannabinoid quantitation in cannabis products using UPLC with UV and mass detection. So I mentioned that I'm actually based in the United States, so I do have this slide entitled Cannabis in North America, but I think some of the topics that I will discuss on this slide are actually relevant globally. But to begin, really, the terminology in this industry can be quite confusing, and that's why I like to start off with this quote that I heard in grad school, which is, "The beginning of wisdom is to call things by their proper names." So I think that's a great place to start here. So throughout this presentation, I will be using the term cannabis quite a bit. I use cannabis as an umbrella term since it's technically the genus of this plant. And there are other terms that may pop up throughout and in the industry anyway that sort of have more specific meanings. So marijuana typically refers to high THC plants. We typically don't use this term as often anymore. Hemp is defined by, in the US, the 2018 Farm Bill, which means that plants that have a THC content less than 0.3% total THC can be defined legally as hemp. Medical cannabis is defined as high THC products that are legal for adult use for medical purposes, which is shown here in this map, highlighted in yellow. Those are states where medical cannabis programs exist. And then adult use is the revised terminology for what was once called recreational cannabis, which means high THC products that are legal for use and purchasing for those over the age of 18. So this map here, highlighting the United States, you can see that there's kind of a varying distribution of states that have adult use laws, medical use laws, and then some that only have hemp. But this seems to be in flux. And rules are changing by the day, so it can be a bit challenging to keep up with. And these cannabis is regulated by a few different entities within the United States. The USDA, the U.S. Department of Agriculture, regulates hemp and helps to set rules for regulation of hemp, how it's grown, and how hemp typically gets tested for compliance, making sure that THC level is low enough. The DEA, the Drug Enforcement Administration, also plays a role, as some cannabinoids are scheduled and used for pharmaceutical purposes. And then also the FDA has a hand in regulating these as well, as many different products exist on the market that sort of fill in the space of being a dietary supplement, although these products technically by FDA rules are not dietary supplements. So just wanted to point out that these three different agencies really have a hand in regulating cannabis products as a whole in the United States, and that I'm sure in other places in the world they might be regulated a little bit differently. The cannabis market is certainly not experiencing a shortage of innovation. Every day new products hit the market, like new types of flour, different types of foods, gummies, oils, beverages. But despite how different all of these things are, there's one thing that they have in common. It's that they all need to be characterized. For instance, what's in this product, which cannabinoids, and how much of them are there? There are many different chemical constituents that are found within cannabis. Here within this presentation, I'm specifically going to focus on cannabinoids. But one of the reasons why analytical testing can be challenging is because of how complex the plants are and how complex it is to analyze for your analytes of interest when they're mixed in with other things like foods, beverages, and beauty products. I want to briefly walk you through the life of a cannabis plant from the field to the shelf to highlight when cannabinoid testing can be useful. First, when you're growing and harvesting your cannabis plants, it's important to consider whether or not your plants actually match the phenotype that you desire and whether or not your hemp crops are compliant with state and federal regulations. Next, when you're processing these plants to create other products, it would be really helpful to ensure that your extraction efficiency is what you expect, and that if you're creating different types of extracts or isolates, that they're as pure as you think they are. Next, It can also be really helpful to test for cannabinoids when you're formulating new products. It's not just about trusting the input, it's about verifying the output. Now, as you prepare to label your products, it's important to make sure that the label you're putting on your product actually matches the product that's in the bottle. And last but not least, it's also helpful to check for stability. This can ensure that you are characterizing the lifetime of your product and that the labels that you put on there will truly match the product in the bottle 6 months or 1 year down the road. The most commonly sought after cannabinoids are THC, THCA, CBD, CBDA, and CBN, but it can also be helpful to characterize the content of other cannabinoids in your samples. For example, Delta-8 THC is a hot topic recently, and CBG too. Already having a method that can detect them means that you are already ahead of the game. The method that I'm presenting to you today focuses on the 18 cannabinoids shown here in this table. So Waters takes pride in creating and offering total analytical workflow solutions in order to make our clients' lives just a little bit easier. We directly provide the analytical technologies like the UPLCs and their detectors. We can provide the columns, the different sample preparation chemistries, a method, and the processing tools that are needed to effectively characterize and quantitate your analytes of interest in your samples. And at the end of the day, we provide you with a software that can help you manage your data in a compliant manner. The first step of the process is preparing your samples. For instance, flour. We use the Specs Geno grinder in our Waters laboratories to help facilitate this process, but you could also use something like a handheld coffee grinder to grind up your samples, as long as you choose to make sure that it is consistent every time you do it. So to begin, you would take half a gram of your flour sample, place it into this container with 2 steel grinding balls and 20 milliliters of acetonitrile. You then simply just process for 3 minutes, sonicate that extraction solution for 20 minutes, centrifuge for 5 minutes to make sure all the particulate matter ends up at the bottom of the vial. You would then filter with a 0.2 micron PTFE acrodisc filter, and then you could dilute your samples 80 and 2,000 times to make sure that they are ready for analysis. Next, we have edibles. The example that I'm going to use today is gummies. It is very helpful to process these gummies using the Specs freezer mill, as they are ground up under cold temperatures to keep them from getting sticky. This sample is slightly different, but just as easy. You would take 1 gram of your ground up samples, mix them with water, and then make sure they're in solution by vortexing and sonicating. Then to that solution, you would add some acetonitrile and our CEN catcher salts, which are shown in the silver packet on this slide. You would then shake that mixture for 1 minute, centrifuge for 5 minutes, and then you will see that there are 2 layers in your 50 ml tube. The layer on the top is acetonitrile, and you can directly suck some of that up, put it into a vial, and analyze. Infused beverage preparation is also fairly easy. You would only take 10 mils of your beverage plus 10 mils of acetonitrile, put it into a tube, vortex for a minute, and then you would also add the CEN catcher salts there. You would then shake and centrifuge, and then the acetonitrile layer that will end up on the top is what you would directly put into your autosampler vial for analysis. To ensure and show that the method that we created does actually have suitable percent recoveries, we decided to do a spiking study. And this spiking study is very similar to that of the ground gummy preparation that I talked about previously. But this time, with the ground gummy material, we added a spiked solution of 18 cannabinoids mixed in with the homogenized gummies that were predetermined to contain a certain concentration of CBD. The final added concentration of these extra cannabinoids was 0.005%. We then prepared this mixture the same way as previously described by shaking and doing a liquid-liquid extraction with the CEN catcher salts. And then we diluted the sample to make a final volume of 1.1 milliliters, and we called this our pre-spiked sample. For the other sample, we chose to take our ground gummies and prepare them as previously described. We then took that acetonitrile layer, 1 milliliter of it, and added the spiked cannabinoids in after the extraction was already complete to create the same concentration as our pre-spiked sample, but this time, this one is our post-spiked sample. We then used the analysis from both of these samples, did calculations compared to the standard curve, and used that to determine the percent recovery. The method that I'm here to describe today uses both our Acuity H-class UPLC coupled to our PDA detector, which is standard. A lot of methods within the literature and in the field use PDA or UV detection to detect and analyze and quantify the different cannabinoids. But one of the features of this method here is that we also incorporate mass detection. So down below on this slide, you can see our Acuity QDA. We call this a mass detector because it essentially fits within the LC stack that many chromatographers already use, and it doesn't require any special expertise in order to operate. So using both of these detectors, we get quite a bit of data. So what I'm showing you here is an example of what you would see in the Empower software, which is what we use to operate our instruments and edit data. So what you're seeing here is actually a comparison of a few different chromatograms. The top chromatogram is the UV spectra scanning from 210 to 400 nanometers. And as you can see the 18 different cannabinoids there. Down below are those same peaks, but they were detected using mass spectrometry or the Acuity QDA mass detector. One of the benefits of using both of these detector systems in order to evaluate your samples is that you get two pieces of data that help you to determine that this is indeed the compound that I expect it to be. Here I am showing some chromatograms of the lowest calibration standard tested at 228 nanometers for the UV detector at 3.15 micrograms per mil, and for the individual selected ion recording experiments, which scans for a desired mass to charge ratio for each cannabinoid. The mass data was collected at 0.4 micrograms per mil, which indicates here, by also showing the chromatograms, that we were able to achieve lower detection limits using our mass detector versus our UV detector. This is an example of what you would see within the Empower 3 chromatography data system that you would use to control your instrument and to visualize your data. So here you can see 3 chromatograms. The top chromatogram is the UV trace at 228 nanometers, and you can see here that we have 2 peaks highlighted, CBD-A and CBD. Down below, you can confirm that those are indeed the peaks that you think they are by looking at the mass spectrometry data, where in the middle chromatogram, you see that this is QDA data in the positive polarity, confirming the mass to charge ratio of 315 for the CBD peak, and then on the bottom, in the negative polarity, a mass to charge ratio of 357 for the CBD-A peak. Within the Empower software, you can also quickly compare the UV and the MS spectra to each other by this top panel, where it shows the UV trace for CBD-A individually and the specific mass peak of those analytes of interest. Here we show the recoveries of the 18 cannabinoids that were spiked pre and post extraction into a gummy at 0.005%. This top chromatogram on the top right shows the chromatogram based upon the UV data. 5 micrograms per mil was the concentration ultimately that was spiked into this gummy, and that is pretty close to the detection limit of the UV detector. So as you can see, the peaks that are contained within this chromatogram are pretty small. Down on the bottom though are those same samples, but analyzed via the QDA, the mass detector, and as you can see, there is a higher signal intensity for these peaks. That just further affirms that the mass detection works well for these samples at a lower concentration range. Now for the percent recovery evaluation for the PDA versus the QDA. The PDA data can be visualized by the blue bars, and the QDA is represented by the orange bars. Here you can see that the recovery of the cannabinoids and analyzed with the PDA ranged in recovery from 60% to 125%. The recoveries for the cannabinoids analyzed with the QDA range from 94% to 106%, indicating that you have improved accuracy of quantitation using the QDA versus the UV method. Now that we've determined that the method created is suitable and works reproducibly, we can move on to some consumer product data. So this chart that I'm showing here is for 6 different flower samples that were analyzed using this method, and there's a lot of data here. So just as a heads up, there are 3 columns of data within each white box. So the first number that you would see is the percent weight, which is the quantity of cannabinoid that was calculated for each sample using this method. In the parentheses is the percent RSD, and then after that is the percent of label claim, with the label claim being shown in the blue columns. So very quickly we can notice a few things. So for instance, regarding CBN in these flowers, you can see that there was no CBN detected in any of these flowers as determined by this methodology, despite 3 of the labels indicating that there were trace amounts of CBN present. Another thing to point out here is the percent of label claim data that we were able to collect. So just as an example, I wanted to point out flower 065. For CBD, we actually determined that the percent of label claim for CBD in this sample was 141%. This indicates that there is more CBD found in this sample than what is actually listed on the label. This could be important for consumers, and it could also be important for manufacturers who are trying to ensure that they are truly labeling their products correctly and getting their money's worth for the product that they produce. Also, the same was true for Delta-9 THC in this flower, where there was actually slightly higher, 121% of the label claim. This could also be due to THCA, which is also found in this sample, losing its acid and degrading into Delta-9 THC. Now, regarding some cannabinoids and chocolate analysis, we can see here that I have highlighted Delta-9 THC for chocolate 154, 515, and 695. For both chocolates 154 and 515, which are in the first 2 columns, you can see that both had a percent of label claim as 107%, which is not too bad. For chocolate 695, though, they had a slightly higher value at 111% of label claim. Not nearly as bad as some of what we saw with the flower results, but something to keep in mind. For gummies, on the other hand, gummies 868, 632, and 941 were sort of all over the place, with gummy 868 only having 36% of its label claim, while gummy 632 had 111% of label claim. It would be advantageous for these manufacturers of their gummies, and for just gummies in general, to keep this in mind as they formulate products and to consider cannabinoid testing along the way. Now for beverages, we had a little bit better data. Only CBD was found in any of these samples, which was indicated on the label. And for these 3 different beverages that were tested, which did have slightly lower levels than the other products that we tested, all of these matched 100% of their label claim. One convenient feature of the Empower 3 software that you would use to control your LC and do data management is the ability to create custom reports and C of A's. So what I'm showing here is an example of what a C of A could look like for cannabinoid analysis. As you can see on the right side of the page, there's a lot of data there where you can see who the data were acquired by, when the sample was ran, when the sample was processed, and how long the runtime was. You can also choose to include a picture of a chromatogram if you so wish. And one important feature that I'd like to highlight is that you can incorporate custom calculations. Total THC is something that's very relevant and interesting to the hemp market, especially for THC compliance testing. And in order to do the total THC measurement, you have to do a little bit of algebra, right? So we made it easy by allowing you to do these calculations automatically so that you don't have to take data out of Empower and then put it back in when you create your C of A's. Overall, we learned a few things by working through this study. We found that we do have an effective and robust and reproducible UPLC, PDA, and QDA method that can separate out the 18 cannabinoids in under 10 minutes. We did find that the QDA data is better for doing more sensitive measurements and it's more reproducible, but having both PDA and QDA data can be advantageous. The Empower 3 software that controls the instrument and also processes the data is very useful in that you can view all of the data all in one location, and you can also use it to generate reports and C of A's. Next, the sample preparation techniques that we showed you here only vary slightly from sample to sample, but we did find that the catcher salts do work well for doing cannabinoid analysis for edibles and beverages. The percent recovery again shows that the QDA has greater accuracy, but again, both can be useful when used together. We did find a wide range of label claim values, anywhere ranging from 36 to 125% of what was on the label for edibles and for flower. So just another reason why doing cannabinoid testing along the way can really benefit our manufacturers and processors. But on the bright side, the infused beverages that we tested did contain 100% of their label claims. Here are a few final thoughts for you to consider. As this market continues to grow and people continue to innovate and create new products, make sure that you're doing cannabinoid testing along the way. People hope and trust that the labels that are on their products truly match the products that are on the inside. So do your due diligence and take care of your customers. Make sure your cannabinoid content is appropriate because your brand and your reputation depends upon it. And if you do want to do cannabinoid testing, whether you're a third-party testing provider or you're somebody who wants to check in-house, Waters has a solution for you. And we don't just want to drop a blue box off and say, see you never. We want to partner with you. We want to make sure that you have SOPs. We want to provide you with education and assistance because our job is to help make sure that you're comfortable and confident in your analyses. We're not just a vendor. We want to be your partner in analytical excellence. Although I am so happy to have presented this to you today, the real credit actually goes to Kim Van Tran, who is one of our scientists that works at headquarters in Massachusetts. She's the one who actually did all of the legwork and created this methodology and is the reason why I'm presenting this today. I also want to thank Marion Tuhig, who's also an excellent contributor and cannabis advocate at our Waters headquarters. And I also have to thank Christopher Hudala, who is our partner and friend at ProVerde Laboratories. Thank you all so much for your time and attention.
Okay, thank you very much for your attention. We are ready here to answer to any questions you have. Before and give you the time to prepare the questions, I have to make some comments for this presentation. As you can see, it's very important to have the correct flow during the analysis. So that's why the series of this presentation was to show, first of all, to you how the sample preparation is done, and then how the analysis is done. In order to, if we want to measure potency to our samples with high repeatability and in an automated way, we have to use automated solutions. Waters can provide us with the instruments and with the software to achieve these analysis results. It's very important also that we are able to measure cannabinoids every day in the lab, checking all phases of cultivation. We can measure cannabinoids during drying and to see and to determine if there are any affections of drying procedure to our cannabinoids. Because as you know, drying, we can lose even 25% of our THC if drying conditions are not correct. So since you have this equipment, you are able to measure your extractor capability and even to be able to decide and change the extraction parameters. This is what we're trying to show. I don't know if there are any questions from your side. You need an answer, or you can send it to us anytime. We will give you some time to see if there are any questions, and we will start the next presentation in 2:30 CET sharp. So any questions you have for this presentation or for the next presentation can be sent to the platform. There's a question. There is a question saying, if I can't have access to all of the 18 cannabinoids in pure form to create standard care for each, is it possible for me to express quantitative results? If so, how many of these cannabinoids? You can use standards. There are companies in Europe selling standards for this. And we as Hellamco, we have the whole package. Our service engineers and our application engineers are coming to your site, suggesting what standards we have to buy, and we set the method to the instrument, and we show the customer, the user, how they can calibrate the instrument and measure cannabinoids with high sensitivity in quantitation. You can reduce the solvents and the material needed, as there is a question here, for the extraction method now. Is something you can do. The question says if we can reduce proportionally the solvents and the materials needed for the extraction method. This can be decided only after the use of the instrument, after the use of the extraction method you are having. The edge has inside stored extraction methods. You can use that or play with that and decide if you have to use another extraction method or not. Also, there's another question if we can detect some impurities on foreign matter if we use the techniques you showed us a while ago. Impurities, you can check impurities only by using an NMS detector. It's difficult to check impurities with just an HPLC. So QTA, a mass detector, will be used to check impurities. Also, you can check impurities if you are having equipment to measure also pesticides, which it's an MSMS, it's the next presentation. I don't know if it's clear. Generally, this setup HPLC with UV or PDA detector is used to measure cannabinoids. This is the main measurement you can do with that. For the other measurements, you have to use more dedicated and more sensitive detectors. Just for your information, you have to run the HPLC only in isocratic mode. Isocratic mode, 18 cannabinoids, the analysis is done. If there are no more questions, we can connect again in 10 minutes and also answer to the questions that will appear after that. Good evening again. Now we'll move to the next presentation from Waters Corporation. It's Mr. Gordon Fujimoto. He's the leader of Chemical Analysis Department of Water. And the presentation will provide to us the solutions that are used to measure mycotoxins and pesticides in pharmaceutical cannabis samples. It's recorded again. So in any case there are any questions and I'm able to answer them, I will do it after the finish of the presentation. Otherwise, we can collect them and send you an answer.
Hello, thank you for attending our meeting on cannabis quality and safety. In this part, I would like to discuss with you the safety part, in particular testing for pesticides and mycotoxins. Currently in the United States, there are no national pesticide regulations for cannabis that apply in all of the states. There are national pesticide regulations for Canada, but in the US, each state is deciding on their own which pesticides to test for and what the tolerance levels will be for their pesticides. So in the US, there are almost 50 different sets of regulations. Here is an example of some of the varying pesticide regulations in some states and regions. In Canada, they require testing for 95 pesticides with tolerance levels ranging from 10 ppb to 500 ppb, while in Maryland, only 48 pesticides are listed with levels ranging from 100 ppb to 1,000 ppb. California has one of the larger lists in the U.S. Massachusetts requires testing on 9 pesticides, though that number is going to go up, and while it's a small number of pesticides, their tolerance levels for some of them are lower than those used in California. In some states, the regulations are still evolving, as in compounds are still being added to their lists, like in Massachusetts, and tolerance levels are being adjusted. Developing a methodology to cover a very wide scope of compounds with the requirement that the LOQs that must be achieved for them can vary over a wide range can be challenging. That's where standard setting organizations play a role. For our method, we chose to meet the requirements set by AOAC International. Through its working groups, AOAC publishes standardized analytical methods with the aim to increase confidence in the results produced by these laboratories. Government agencies and other organizations will often require laboratories to use AOAC methods. AOAC also publishes standard method performance requirements, SMPRs. These requirements are agreed upon and set by their working groups and are often used as criteria for method validation in laboratories. For pesticides and cannabis, the working group studying this area chose the lowest tolerance level for Canada or a U.S. state as the action level. This resulted in a list of 104 compounds with limits ranging from 10 ppb to 2 ppm. When one ventures out on the adventure of method development, it is a great idea to consider the chemical properties of your analytes of interest. Most pesticides are neutral or very weakly acidic, with partition coefficients or log p-values between 1 and 5. Log p-values can be thought of as a measure of how hydrophobic or how hydrophilic an analyte is, or how readily a compound will partition between an organic phase versus an aqueous phase. Log p-values influence how sample extraction and cleanup procedures, as well as the chromatographic separation methods should be selected. But of course there are exceptions to the norm. Diminocide is a plant growth regulator with a log p-value of -3, meaning it's very hydrophilic. It is also acidic. When primary and secondary amine or PSA absorbents are used for sample cleanup, diminocide is retained on the absorbent, resulting in a reduced recovery. This also happens with orocotoxin A, which is also acidic, although it has a log p-value that is more in the neutral range. Then we have acequinacel, included in the same analysis, which is highly hydrophobic. This makes for a tough analyte list to measure in terms of finding a universal and effective sample preparation method and a cleanup protocol to go along with a suitable chromatographic method. We also have to consider the detection of the compounds. One way to effectively approach a list of chemically differing analytes of interest is to use 2 different chromatographic techniques. In our case, this is liquid chromatography and gas chromatography. Some analytes ionize and are detected much more effectively using one technique versus another, while others work well using either. For example, atrazine ionizes effectively by LC and GC. Imidacloprid, on the other hand, requires LC. Pyrethrins are best detected and quantified using GC. Also, some analytes experience more or less matrix effects in one protocol versus another. For this study, in which we targeted the pesticides listed in the AOAC SMPR for pesticides in cannabis, we developed an overall analysis method in which 75 compounds were analyzed by LC and 30 were analyzed using GC. One thing to keep in mind while looking at pesticide lists and numbers is that some compounds have isomeric forms like purethins. This can lead to an apparent discrepancies in the total number of compounds investigated. Cannabis contains cannabinoids, but we are interested here in pesticides. So why in a pesticide analysis do we care about the cannabinoids? Because they can interfere with the ionization and detection of the pesticides that we are interested in. Now, why are cannabinoids major interferences? The answer is because they are present in high abundance. The reference shown at the bottom is highly cited and provides details. Shown here are the full scan data from positive and negative electrospray analyses, and in the regions highlighted, the peaks in these regions are from various cannabinoids. Note that for GCMS analyses, terpenes are also present in high abundance and may be an issue. The upper chromatogram shows our full scan analysis data again. The bottom scan show the MRM chromatograms for the pesticides we are trying to analyze for. In the TIC for the full scan electrospray data shown at the top, many of the peaks are so intense that they are close to saturating the detector. More importantly, while the mass spec can discern the difference between a cannabinoid and a pesticide, the cannabinoids are in such high abundance that they consume much of the charge available to form ions when they elute and interfere with the ionization and hence the detection of the pesticides we are interested in. The pesticides that elute in the highlighted region, and there are greater than 15 of them, have chemical properties that are similar to those of the cannabinoids. This causes these pesticides to elute with similar retention times as the cannabinoids. Also, because they are chemically similar, they will both be extracted from cannabis in our extraction process. An extraction process that efficiently extracts the pesticides for analysis will also extract the cannabinoids. This means a sample cleanup step will also be needed. It is just not the cannabinoids we have to worry about. Here are some other possible interferences that can be present in cannabis extracts. Cannabis, not surprisingly, contains other components that are common to most plants. Phospholipids can cause problems as they build up on the injector mechanism of your LC or GC, and they can also suppress electrospray ionization. Fats can precipitate out of samples when they are eluted for LC analysis. Not a good thing to be injecting chunks of fat. There is potential for chlorophyll, and if it's not removed, it will reduce the lifetime of your GC column and could contaminate your GC injector. The general objective is to have a sample prep method that is the simplest, fastest, and produces a clean enough sample for your analysis. This is summarized here. As you move down the list, you get cleaner samples, but an increase in complexity and hence cost. We found an acetonitrile-based extraction method similar to that used in catchers, followed by a dispersive solid phase extraction step works well for the preparation of cannabis samples for pesticide analysis. Here's a summary of the sample preparation method. We start with an acetonitrile extraction that is summarized on the left. A system similar to a genogrinder can be used as it pulverizes and extracts the samples in one step. After the acetonitrile extraction, a solid phase extraction cleanup step is performed to further clean up the sample. For the cleanup, 1 aliquot of the extract is sent through a pass-through solid phase extraction cartridge. We use an Oasis Prime HLB cartridge for LC amenable pesticides. Another aliquot undergoes dispersive SPE for GC amenable pesticides. The Oasis HLB cartridge uses a reverse phase mechanism and remove lipids, fats, and some of the chlorophyll, as well as the terpenes. For the GC samples, there is a carbon C18 component, as well as primary secondary amine, a salt, magnesium sulfate, and some graphitized carbon, which removes the chlorophyll and other pigments. Here's a closer look at the cleanup step used to prepare a sample for LCMS analysis. 0.25 ml of water is added to 2.25 ml of the acetonitrile extract. The water aids in the recovery of spinosads. 0.275 ml of this mixture is passed through the cartridge and discarded. This pacifies the cartridge. The rest, 1.75 ml, is passed through the same cartridge and collected for analysis. These steps can be done by gently pushing the sample through the cartridge and using a syringe, as shown at the top, or pulled through the cartridge using a light vacuum on a vacuum manifold, as shown at the bottom. The goal is not to have this sample pass too quickly through the cartridge to avoid drawing oils and fats through the cartridge. This results in a 1:20 dilution. For GCMS analysis, A dispersive SPE process is used. The materials listed at the top typically come in a premixed form and are sometimes already in the centrifuge tube. 900 microliters of an acetone hexane solvent mix is added to the tube containing the DSPE mix. Then 100 microliters of the acetonitrile extract from the genome grinder is added. Vortex this, spin it down, and pull out the supernatant. While some of the acidic analytes are removed, this is okay as they will be analyzed using the LCMS method. What is important in this DSP step is that it removes a lot of the fats, phospholipids, chlorophyll, and a major chunk of the cannabinoids that can also interfere with the GCMS analysis. An example of how this can clean up a sample is shown on the right. Once the sample prep has been done, then it's time for analysis. Shown here is a water system that can be used for pesticide analysis. A unique feature of the water systems is the universal ion source, highlighted in orange. This allows for the use of one mass spec for both LC and GC analyses. The switch from LC to GC is fast and easy and is an attractive option for those who are starting up labs or building capacity with limited funds. At waters, we have atmospheric pressure ionization GC, which we call APGC, which operates under ambient pressure conditions. This produces a softer ionization mechanism for compounds, which results in more intense precursor ions, making it a very sensitive measurement technique. Overall, it is a great option for labs looking to reduce expenditures without sacrificing capabilities. A master method was developed to analyze for 105 compounds, as mentioned before. This covers the list of pesticides for the states in the US and in Canada. Cannabis regulations often require analysis for mycotoxins along with the pesticides, and they are often included in the list of pesticides that must be analyzed for. We can see how we have divided up the compounds between LCMS analysis and a GCMS analysis to cover the wide range of compounds we are interested in. Details on this are available in an application note. The optimum MRM transitions and the optimum cone voltages and collision energies for these transitions have been worked out for the pesticides and mycotoxins in this method. Shown here are the MRM parameters for some of the compounds analyzed by LCMS-MS. A full listing of the parameters for all the compounds is given in an application note describing this method. So the mass spec parameters for the cannabis pesticides have already been worked out for you. Similarly, a full listing of the parameters for all the GCMS compounds is also given in the application note. MRM parameters are shown here for some of the compounds. The LC method has been worked out for the LCMS-MS analysis is summarized here. It utilizes a methanol gradient with ammonium formate and formic acid for additives and has a gradient that runs from 2% methanol up to 99% methanol to be able to handle both the polar and non-polar compounds we have to analyze for. The rest of the parameters are standard UPLC parameters, so nothing exotic. Similarly, here is a GC method used for APGC analyses. This is a standard GC method. No exotic GC settings are needed to perform APGC analyses. GC methods that have been worked out well for you in other GC-based analyses, such as a GC-EI analysis, can be used without modification with APGC. You have the added advantage that you can also use higher carrier gas flow rates or switch to using nitrogen as a carrier gas without having to make adjustments to the mass spec that can be needed if you are doing a GC-EI analysis. Now, building a method for 100 or so compounds can be a very involved process. We use Quantpedia as a tool to enable rapid automated setup of methods with automatically scheduling MRM transitions to maximize performance. A Quantpedia library is available for the cannabis pesticide analysis, so you do not have to type in the parameters for 100 compounds. It allows the user to generate the LC or GC method, the mass spec MRM method, and the software processing quantitation method in only 3 steps. Step 1, select run samples from the main Quantpedia menu. Step 2, select the analysis you want to run from the list. I know it's hard to read, but in this example, the California pesticide analysis has been selected. Step 3, enter the name you want assigned to the methods you want created and indicate where the created methods are to be stored. You can choose to have all the needed methods created or just some of them. Many users will want all of them, but maybe, for example, you want just the method with the mass spec parameters. You do not have to use Quantpedia every time you run your pesticide analyses. as once created, you can use the methods over and over again. Quantpedia does provide a means for central storage for your methods. If your lab runs a variety of analyses, you can go get the methods you need for your current study without having to go searching through old data. Having a detailed, tailored acquisition method along with a comprehensive processing method will help implement your overall analysis faster and with fewer errors. There's a complete suite of software tools to aid you in implementing pesticide methods in your lab, along with Quantpedia, which contains a library of methods already worked out. The IntelliStart and QuantOptimize tools are available to help you determine the optimum mass spec parameters for any new compounds that you may be required to analyze for that are not already in Quantpedia. Let's look at some results. The cannabinoids that will be shown are from analysis of a 10 ppb spike into matrix. Shown here are some of the GC-MS-MS analyses. The GC-MS analysis cal curves shown here have an R-squared value greater than 0.995 and cover the range from 100 to 500 ppb. The first example is from a fungicide, azoxystrobin. This GC-MS-MS example is for bensulfion, which is an insecticide and a nematicide. Shown here are examples of recoveries from the compounds analyzed for by GC-MS-MS in samples spiked to 3 different concentrations. The analyte lists are long and the recovery slide is difficult to read, but this gives you an idea of the recoveries that can be achieved using the methods described here. It is not possible to fit the data from all the compounds onto one slide, but all of the data are published in an application note and is freely available. Shown here are some of the LC-MS-MS results. The LC-MS-MS cal curves shown here have R-squared values greater than 0.995 and cover the range from 1 to 2000 ppb. For the first example is for spiraclofen, which is an insecticide and an acaricide. This LC-MS-MS example is from etoxazole, which is an acaricide and it can be used as an insecticide too. This is for the LC-MS analysis for alpha toxin B2, one of the mycotoxins that have to be analyzed for in some of the areas. Shown here are examples of recoveries for the compounds analyzed by LC-MS-MS. Again, the analyte lists are long and the recovery slide is difficult to read, but this gives you an idea of the recoveries that can be achieved using the methods described here. Again, it is not possible to fit the data from all the compounds onto one slide. Like the data on the GC-MS-MS compounds, all the data on the LC-MS-MS compounds are published in an application note which is freely available. Here we see the results of the AOAC residue testing method. Up to 105 compounds in the method, 99 have recoveries between 70 and 120%. 4 compounds had recoveries between 60 and 70%, and while the recoveries are a bit low, the LOQs achieved were better than the limit needed for regulatory action for these analytes. The remaining 2 compounds degraded quickly and significantly after extraction, leading to their poor recovery. For these 2, first find out whether they are needed in your area, and if so, modifications of the extraction method can be made. Full details of the method can be found in the application note. In conclusion, a method that includes extraction, cleanup, and analysis by a combination of LC-MS-MS and GC-MS-MS has been shown to meet the AOAC SMPR for pesticides in cannabis. It has good coverage of the pesticides that have to be analyzed for in cannabis and can be used as a basis for an analytical method in the state or region that you are working in. We have published a variety of cannabis pesticide methods which are shown here. Since we have limited time today, I have focused on the AOAC methodology, but please keep in mind that we do have specific methods for California, Oregon, and Canada. The AOAC method application note, shown at the rightmost part of the slide, is the most comprehensive method that we have developed and meets the regional pesticide testing requirements in the US states and in Canada. You can find these application notes on our website, or you can email us and we would be happy to provide them for you. And finally, I'd like to acknowledge the work of the scientists at Waters along with Chris Udala at Proverdi Labs who have worked on the development of the methods described here. And thank you.
Okay, thank you very much for your attention again. You have time to provide some questions if you have. In the meanwhile, I have to highlight some of the parts of the presentation. First of all, it's very innovative. This solution is provided from Waters for a hybrid system, a system that has only one MSMS detector coupled with 2 inlets. So you can have one system working as LC-MSMS or ACDC-MSMS, not simultaneously. You just have to plug and unplug the source. It's 1 minute you have to spend about that. But it's very important because you have to validate only one instrument, one software, because MassLynx is a software that will be used to run both instruments. You have to have only one nitrogen generator to run the system. And since the samples that you have to analyze in the cannabis industry are not so many samples per year, you don't need to have two dedicated analyzers running simultaneously samples every day. You can have this system as a choice in your lab. Also, as I can see here, there's a question about softwares. All these softwares that are mentioned from Waters are softwares that are coming in the basic package of MassLynx. MassLynx is the core software of Waters and has the options of IntelliStar, Quampedia, and Radar. This extra softwares is the software that add more value to the system. More or less, all the systems in the market are having decent resolution, decent hardware. The huge difference is the software, because at the end of the day, what makes the analyzers' life easier is to have a nice software and be able to validate it and rely on the software. Except this equipment that we presented to you for analytical measurements, there are also another big list of equipment that need to be to a quality control cannabis lab. We have to have definitely chambers to measure product stability, chambers that have stable condition in moisture and temperature. You have to have balances based on the GMP requirements. You have definitely to have moisture analyzers in order to be able to check your drying rooms every day. Besides that, you must be also able to provide microbiological analysis and be able to control several microbiological parameters in your samples. And another comment about pesticide is that the lists that are provided from Waters or other companies in the field are based usually on a USA list of pesticides, because all these instruments are mainly manufactured in USA. According to European Pharmacopoeia for natural drugs, we have to check 69 different pesticides. Most of them are measured with LC-MSMS, but there is a list of 6 to 8 of them that are measured only by GC-MSMS. This list of pesticides is fully different than the lists that are in the States, because in Europe, we use different pesticides that they use in the States. So, Hellamco, and I make a parenthesis here, I'm excited to inform you we have signed our first contract to fully equip a quality control cannabis lab in North Greece. We will be able also, except providing the equipment, our application engineers will be able to set the methods and provide to the customer a full package and perform the analysis of pesticides, mycotoxins, terpenes, and cannabinoids. This was from my side. I don't know if you have any more questions about this part. If you have any questions later, you can send it by email. I will be glad to answer it. Now we will move to the next presentation. We will have a presentation for VALOYA. Ms. Stina Kotiranda is the presenter. VALOYA is a company providing LED lights and solutions for cannabis cultivation. I welcome Stina to our webinar, and she will be able to provide you a presentation regarding the effect of light spectrum on cannabis morphology and cannabinoid content. Thank you very much.
Yes, thank you. Let me see. Yes, I hope you can now see the presentation. Thank you for the introduction. So my name is Stina Kotiranta. I'm from VALOYA, Finland. VALOYA is an LED company, and I've been doing plant and LED related research for the past 10 years. And my today's topic is about how we can modify the cannabis morphology, so the growth appearance, and the cannabinoid concentration with different light spectra. So here's the content of my presentation. I will start with a short introduction to VALOYA, our research history, and give you a short photobiological intro so that you can better follow the second part of the presentation, where I will share some research results from our in-house experiments and some experiments that we've done together with universities and research institutes regarding the effect of UVA and infrared wavelengths on cannabis yield and cannabinoid content. Then finally, I will just show one slide of our technology and how our customers use our products. So if we start with the introduction to the company, so we are a relatively old LED company. VALOYA was founded in 2009, and we started doing research from the very beginning. And this graph on the right-hand side shows the cumulative values of research that we have conducted each year. So up until last year, we had conducted more than 600 plant experiments with more than 300 species or varieties. And we started doing cannabis research in 2014. And until today, we have conducted more than 40 experiments with cannabis. And we do cannabis research in-house, but we also have research partners in USA, Spain, Italy, Austria, Finland, and Germany. Then the short photobiological introduction. So we can talk about photosynthesis and light signaling when talking about light. I think everybody knows that light contains energy, and the energy is in the form of photons. And these photons are absorbed into the leaves by different pigments. The most commonly known is the chlorophyll pigment, the green one. And the photon's energy is converted in the leaf into chemical energy in the photosynthesis process. So where the plant creates sugars and then biomass is created from those sugars. But light also contains information about the environment to the plant. So the plant kind of sees different light colors and detects these colors through photoreceptors. And depending on the light color the plant sees, different morphological and developmental and chemical responses happen in the plant. And in the next slide, I will go through the different light colors in a growth spectrum and what do the different colors of light do for a plant. And I will use cannabis as an example plant here. So here in the middle of the slide, we have a VALOYA spectrum called Sol-Ray 385 presented. And as you can see, the spectrum is a continuous spectrum. So it contains all of the wavelengths from the UVA end to the far red end of the spectrum. And now I will go one by one each of these color ranges to explain what do these colors of light do for cannabis plant. So if we start with the UVA, so that would be radiation below 400 nanometers. These photons contain a lot of energy. So when a plant detects photons coming from this wavelength area, it starts to protect itself from this high irradiation energy, and it stimulates different compound accumulation, such as cannabinoids and terpenes. It also helps to make the plants more pest resistant. And then we have blue light here. Blue light also contains a lot of energy. Generally, blue light makes the plants more compact, so the stems become shorter. blue light has similar effects to UVA light. Blue light also stimulates cannabinoid accumulation in the flowers. Then in the middle of the spectrum, we have a big green portion. So green is something that makes this particular spectrum appear white to the human eye. Green light also penetrates the leaves more efficiently than blue light or red light. So the lower layers of the leaves can utilize green light for photosynthesis. Also, the lower leaves in the canopy can use green light for photosynthesis. Then we have red light. Red would be the highest peak in this spectrum, as it is in most horticultural spectra, because red light is the main component needed to drive the photosynthesis process. So the more there is red light, the higher the biomass accumulation is theoretically. Red light is also included in the photoperiodism process, which is of course important when talking about cannabis, which is a short day plant. Then we have far red at the end of the spectrum. Far red light increases the photosynthetic area, meaning that far red light makes the individual leaves bigger, so there's more leaf surface area to capture light. Far red light also increases the plant height, so it makes the plants taller and decreases the branching. Far red light has also been shown to decrease some of the secondary metabolites in cannabis, and I will shortly show you some examples. So now you know the basics of a spectrum. So what do each of these wavelengths do? So I hope you understand that it's complicated to create a growth spectrum. You have to know what each of these wavelength areas do for the plant. So when we design a growth spectrum, we have to take into consideration who is it for and what is it used for. And at VALOYA, we try to have the best possible spectrum for each growth phases for each plants. And here I have presented the cannabis cultivation cycle in a simple format. So normally people start from mother plants, and then they multiply their plants either through micropropagation or traditional cloning methods. And for example, in micropropagation, our customers are looking for higher multiplication rates and quick root formation in the end and vigorous seedlings. Whereas in traditional cloning, they want to have a quicker root formation and a lot of roots, and ensure that the seedlings are in good condition when they are transplanted into bigger pots. In today's presentation, I will concentrate on the flowering part. So all of my examples are about how to affect the yield, how to affect the cannabinoid content, and how to affect the terpene content by changing the light spectra. So let's talk about flowering experiments. I decided to take 4 experiment examples into this presentation. The first one will be about the effect of UVA on flower yield and cannabinoid and terpene content. The second example is about far red. How does far red affect the morphology and the cannabinoid and terpene content? And then the third and fourth examples are more applied research, where we compared LED spectra versus a ceramic metal halide or a high-pressure light source. So let's start with the UVA. In the UVA experiment, we had 2 light treatments. So we had the VALOYA Solray 385 as a control treatment. So this is the same spectrum I showed you in the beginning, where I explained all of these wavelength areas. So here we have the UVA component, high blue peak, a lot of green, a high red peak, and a far red bump at the end. The second treatment, I'm calling it Solray 385 minus UVA. So this is the exact same spectrum, but we have just removed the UVA part in this spectrum. So the light levels are the same, but just the UVA is missing in the second treatment. And this experiment was done together with the University of Helsinki, Natural Resources Institute of Finland, and Kekkilä PVP. So let's take a look at the the trial setup first. So very clear, two treatments, so two spectra were tested. We had 6 experimental plots. So both spectra were replicated 3 times in the experiment. And each of these plots contained 9 plants. And in the end, we measured the flower yield, the cannabinoid content, and the terpene content from the plants. And now we go directly into the results. So let's start with the flower yield. So we measured the yield from 21 plants per treatment. And what we can see from the data graph here is that the flower yield under the sol ray treatment, which has UVA, was 14% higher compared to the treatment which did not have UVA. So on the x-axis we have the treatments, and on the y-axis we have the yield dry weight as grams per plant. Then we move on to the cannabinoid results. Here on the x-axis we have different cannabinoid compounds. So we have CBDA, THCA, and THCVA. So as you can see, this was a very CBD dominant variety which we used in this experiment. And without UVA, the CBD content was 11% lower compared to the treatment where we have UVA included. With THCA, the difference was 13%, and with THCVA, it was 47%. Of course, this amount of THCVA and THCA was low in general. But if we look at the main cannabinoid here, the CBDA, we can say that approximately 11% more cannabinoids can be achieved by adding UVA into a spectrum. And these bars are a mean of 45 data points. So we measured 15 plants per treatment, and from each plant we took 3 replicate measurements. And we measured the cannabinoids with the HPLC method. Then we go to the terpene content. So here you see on the x-axis, I've clustered the monoterpenes and sesquiterpenes into individual groups. So again, the orange bar represents the sol ray 385, so the original spectrum, and then the purple bar is the same spectrum without UVA. And I think even clearer now with the monoterpenes compared to the cannabinoids is that the UVA really increases the amount of secondary metabolites. So we could increase the monoterpenes by 32% with the small addition of UVA in the spectrum. With sesquiterpenes, the difference is not that dramatic, indicating that perhaps the spectrum does not have that big of a difference to the sesquiterpenes. But of course, more research can be done. We measured, again, 15 plants and took 3 replicate measurements per plant for this data, and we used the gas chromatography device to measure the terpene content. So that was the first demonstration with the UVA. So now we learned that the UVA increased the yield, increased the cannabinoids, and increased the terpene concentrations. Now we move on to the far red demonstration. So how does addition of far red affect the cannabis yield and cannabinoid and terpene composition? So again, we had the same control treatment, the sol ray 385 to begin with, but as with the UVA where we removed the UV, in the far red demonstration, we actually added far red into the spectrum. So the amount of far red is really high in this this second treatment. So we wanted to see how does the addition of FARED affects the yield and the secondary metabolites. And this experiment was again done together with the University of Helsinki and the Natural Resources Institute of Finland and Kekkilä PVB, who provided the substrates for the experiments. Trial setup, very similar to the UV experiment. Again, 2 treatments, Solray 385 and Solray 385 plus FARED. Each light treatment was replicated 3 times, and again, 9 plants per plot was planted. We measured flower yield, cannabinoids, and the terpenes. And we move on to the flower yield results. Here, before going to the graph, I want you to take a look at the images on the left-hand side. So this plant on the left lower corner has been growing in the FARED enriched environment, whereas this plant has been growing under the Solray 385. The first thing you notice is that these are a lot taller. So this is the 100 centimeter mark, the white line on the background. So these are almost a double the size as these are. And this is due to the FARED. So the FARED causes this elongation of the main stem. What you also notice is that the FARED inhibits branching. So here we have a lot of side branches formed, whereas under the FARED enriched spectrum, there are basically no branches, or they're really short and skinny. And if we look at the main buds, so here's the main bud from a FARED from the FARED treatment. So they are really thin and loose, whereas the main bud under Solray 385 is typically really heavy and dense. And the combination of having a very thin main bud and no branches led to a huge yield decrease under the Solray 385 plus FARED treatment. So as we could not basically collect any side buds from these plants, the overall flower yield remained more than 200% lower compared to the Solray 385. So the amount of FARED in this spectrum was really, really high. Then if we look at how does the FARED affect the flower quality? Here on the x-axis, I have again the compounds of the different cannabinoids. So we have CPDA, CBDVA, and THCA. And the orange bar is the Solray 385, so the control treatment. And then the purple bar is the one where we added FARED. And what we noted from the data was that we got 19% more CPDA, 60% more CPDVA, and 23% more THCA when we did not add FARED into the spectrum. Again, all of these bars are a mean of 45 measurements, and we measured the cannabinoids with the HPLC method. Then we look at the FARED experiment and how does FARED affect the terpenes. Like with the UVA, we saw a huge increase in monoterpenes by adding FARED, by adding UVA. And here it seems that the monoterpenes are quite sensitive to spectral changes. So if we add FARED, we actually can lose 56% of the monoterpenes. With the sesquiterpenes, the difference is not that big. Similar to what we saw with UVA, we couldn't really affect the sesquiterpenes with the UVA. And also the difference here is it's 25%, it's big, but it's not as big as the monoterpene difference. So it seems like the FARED really reduces the amount of terpenes. Again, 15 plants were measured per treatment and 3 replicate measurements, and we use the gas chromatography method to measure the terpenes. So now we have learned that with UVA we can increase cannabinoids, with FARED, we can decrease it. Also with FARED, we can affect the elongation and the branching of the plants. So now we go on to the third and fourth experiment examples. So I'll start with the LED versus ceramic metal halide. And hopefully we can use some of the information that we have learned from the demonstrations of 1 and 2 in the third and fourth results conclusions. So this experiment we did again with the Sol-Ray 385 spectrum. So this is very familiar to everybody already by now. And here is the ceramic metal halide spectrum, a very different one from the LED spectra that we have in the market. What is interesting in this spectrum is that the ratio of all wavelengths to each other is almost equal. But I would say that the main difference between the Sol-Ray 385 and the ceramic metal halide is the amount of FARED. So here we have a lot of stuff at the end of the spectrum, whereas here we don't really have anything, just a small bump. So what we learned from the FARED demonstration is that if we have this much FARED at the end of the spectrum, we can expect elongation of the main stem, maybe slightly less branching, and maybe yield reductions. So let's see what the research team at the University of Hohenheim concluded. So this experiment was done again with 2 spectra, so Sol-Ray 385 and ceramic metal halide. Very familiar setup. So we had 3 replicates of both spectra, and we measured the morphology and the flower yield from the plants. And in this experiment, they used 3 different varieties, and I have images of 2 of them in this presentation. So if we look at the morphological differences between LED and ceramic metal halide grown plants. So on the left-hand side, we have plants. This is one variety which was grown under ceramic metal halide and under Sol-Ray 385. So we see that the main stem was definitely elongated under the ceramic metal halide, and we believe it's because of the high amount of FARED in the spectrum, whereas the branches were almost the same height as the main stem under the Sol-Ray 385. The researchers weighted the dry weight of the branches only, and what they concluded was that the branches' dry weight was actually 16% higher under the Sol-Ray 385. So the individual branches were thicker. There were also more branches under the Sol-Ray 385. But when they measured the main stem dry weight, that was the opposite. So we had 20% less main stem dry weight, indicating that the plants under the ceramic metal halide put most of its energy into growing the main stem and not the side branches. Interesting. And now if we think of the FARED demonstration that I showed you earlier, the reduction in the branching resulted in lower flower yield. This is the second variety that was grown. It's naturally a shorter type of cannabis, but again, we can still see that under the ceramic metal halide, the main stem is more elongated compared to the Sol-Ray grown plants under the ceramic metal halide. So if we look at the flower yield results, the flower yield was 30% higher under the Sol-Ray compared to the ceramic metal halide. And I believe this is due to the lower amount of branches, and that the branches were not as thick as the Sol-Ray branches, so that the branches cannot hold that much flower yield. So that was the third demonstration. And then we will move on to the final research demonstration, where I will present you the results of LED versus high-pressure sodium. So how do these two spectra differ from each other, and what kind of results can be expected if a grower changes from high-pressure sodium into LED? Here are the spectra presented. As you can see, this name is Solray. So this is the previous version of the Solray 385, but it's very similar to what we have seen before. So there is the UVA component, blue, green, red. Here's a high-pressure sodium spectrum. Now, there are big differences between these two spectra for sure. The first one is I want you to pay attention to is the lack of blue and UVA wavelengths in high-pressure sodium. And if you remember from the beginning, blue was the one that makes the plants more compact, and UVA and blue together enhance the cannabinoid concentrations. Also, what is interesting is that there is no peak in the red area, but there is quite a lot of far red in this spectrum. So the red to far red ratio is low in this spectrum. So we can again expect maybe some elongation and weaker branches with this type of spectrum compared to the Solray spectrum. But let's see how do the results look like. So if we look at the trial setup, we had two spectra, and in this experiment we had one plot for both spectra, and we didn't have simultaneous repetitions, but what we did was that we conducted the experiment 6 times in a row, and this research was conducted together with the Italian research institute Creacin. And I will show you the flower yield results and the cannabinoid results from all of the 6 experiments. So we can move on to the results. Again, let's start with the morphological differences between the two spectra. So high-pressure sodium plant versus Solray plant. High-pressure sodium plant is elongated. The branches are significantly shorter compared to the main stem, whereas the Solray creates like a bushy, compact plant. And if we look at the flower yield, so grams per plant, under the high-pressure sodium, the flower yield was 28% lower compared to the Solray treatment. And we believe it's because of the side branches cannot hold that much flower yield. You can see they are thin and the buds are far away from each other, and they're not filling as well as under the Solray spectrum. Also, as the cannabinoid content, I will reveal you already at this point that the cannabinoid content was higher under the Solray. So when we have a higher cannabinoid content together with a higher flower yield, the total cannabinoid yield per plant was significantly higher under the Solray plant. And I think everybody can understand that if we get like 59% more cannabinoids with a certain type of spectrum, it is a really good increase, especially if you are doing extraction from your flowers. So, but let's take a closer look on the cannabinoid concentrations in the 6 experiments. So we got constantly higher cannabinoid concentrations with the Solray compared to high-pressure sodium. And I have 3 graphs on this slide. The first one showing the THC content, the second one showing the CBD content, and the third one showing the CBG content. And in each graph on the x-axis, we have the trial number. Like I said, we did it 6 times. So this is the trial 1, trial 2, 3, 4, 5, and 6 for each graph. And then on the y-axis, we have the concentrations for these compounds. The purple bars in each graph is the high-pressure sodium result, and the orange bar is the Solray result. And as you can see, in each experiment, the Solray's bars are always higher in THC, in CBD, and in CBG. And if we calculate the increase, then in percentage and take a mean of all of the 6 experiments, we noted that the average increase in THC was 22%, CBD was 11%, and with CPG, it was more than double compared to hypersodium. So very significant increases can be achieved by using a good light spectrum during the flowering phase. So this would be my, I have 2 slides left, so the conclusions. I have 3 points that I would like you to remember from my presentations. The first one is UVA and blue light increase the cannabinoid and terpene concentrations. Second point is that far red increases the plant height, but can decrease the cannabinoid and terpene concentrations if you give it a lot. Third point, we can get higher flower yields and higher cannabinoid concentrations by using a spectrum like Solray 385 compared to the traditional lighting sources like ceramic metal halide or hypersodium. If you're interested to read more about the research that we've done, we have published papers. Here are examples. These are available online, and if you cannot find them for some reason, you can always contact me later on. I have talked a lot about spectra today. Just wanted to show you some images of how we deliver these spectra to our customers. In these examples, they are the Solray 385 spectrum. As you can see, it's beautifully white spectrum, so it's easy to inspect the plants underneath this type of spectrum. We have the Solray 385 spectrum in a BX format, so it's a bar-shaped fixture, ideal for multi-layer cultivation. It's also available in this top light model, which is suitable for a greenhouse installation, or also a closed environment where you would grow in a single layer. This image is from California, our customers called WeGrow, and then this image is from Abdera Green Farms from Spain. This is my last slide. I would like you to ask me questions if you have any, and thank you. You can ask me questions now or later on. Contact me, stina.kotiranta@valoya.com, or my colleagues through sales@valoya.com. Thank you.
Kiitos, Stina. Thank you very much. We already have some questions from the audience, and I will start with the question we have. First of all, the first question is regarding your unique spectrum. Would you say that cannabis that is destined for fiber exploitation is better to use red light rather than UV light?
Can you repeat the question?
Regarding your unique spectrum.
Yes.
Would you say that cannabis that is destined for fiber exploitation is better to use red light rather than UV light?
Yes. So for fiber production, I would not perhaps use the UV that much, because that would make the plants more compact. And in fiber production, you would want to have a lot of stem material, definitely.
Okay. Thank you. Also, another question. That the flowers that were examined in your study were fresh or dried in fluorescence?
They were dried. So we dried all of the flower material before doing the cannabinoid and terpene analysis, also before weighing the yield.
Mm-hmm. Okay, it's clear. Also, the final questions, until any other question appears. The plants measured your CMH vs Solray 385. Did they complete their biological cycle? Also, the varieties used in these experiments, were they monoecious or dioecious strains?
So these were all hemp material that we used. So only CBD material was used. and they were not monoecious plants, so only female plants were used.
Yes, they completed also the biological cycle, the plants that were
Yes.
used. Okay. And now another question appeared, and another one. We have questions rolling. On the topic of LED vs HPS, the variety used was a type 1 high in THC.
Yes, so in the experiment series that we did together with the Creachin Institute, we used a high THC variety. It was a variety bred by the Research Institute.
Yes. Also another question, does the direction of the light play any role? For example, what if the lights were fit on the side of the plant too? So the direction of the light is on the top or the side of the plant. What's best?
This is a good question. So plants can sense the direction of light. So I think everybody knows the phenomenon. If you have a sunflower in the field, they turn towards sun during the day, and cannabis would do the same. I advise to have a top light, and if you want to have additional light to that, then you can have interlight similar to tomato, but to have only light coming from the side, I would assume that the plants would start to curve towards the light.
Also, do you think it would be of any use or better results, we would have better results in total if we would change the light spectra during the plant's development stages?
Absolutely, yes. So I think whether you start from micropropagation or from cloning, it is advised to use a specific spectrum during that time. Also, of course, adjust the light intensity for each growth phase. And then during vegetative phase, we would advise to have a spectrum which makes the plant as big as possible, as soon as possible, so that it is ready for the flower phase as soon as possible, and the branches are so strong that they can hold all the flower yield that is coming.
And as we know in cannabis industry, time is money. So the faster the best is the best way to do it. The final question now. Sol ray spectra contains more UVA than Sol ray 385. Is this correct?
The Sol ray contained actually the same amount of UV, but the peak of that chip was a different wavelength area. So the Sol ray peak was at 405 nanometers. So the UV came from the tail that was going to the below 400 nanometers. But the Sol ray 385 has the peak at 385 nanometers. So nearly all of that light is below the 400 nanometers. So the amount of UVA is same, it's just how it is done. It's different.
And the second part of this question, Sol ray should have higher yields than Sol ray 385. Is that correct?
No. So when we made the change from Sol ray to Sol ray 385, we made a lot of comparative experiments, and we wanted to maintain similar flower yields and similar cannabinoid concentrations in the cannabis plants. So the yields are the same.
Yes.
With Sol ray and Sol ray 385.
These were the questions we had for you. And just there are also two questions appeared for the previous part of the presentation. So before we say goodbye to you, let me give you a moment to answer these two questions. And in case any more questions appear for you, we will move to that also. Regarding the analytical lab, there is a question of how many samples we have to analyze per harvest. Depending on the analysis, if we have to analyze pesticides, mycotoxins, and terpenes, has to be one analysis per batch. The batch is defined from your GMP consultants. There are different approaches how to define a batch. Some people define the batch based on the drying procedure. Some people define a batch that it has to do with a greenhouse or a flowering room. this is up to the GMP consultant and the GMP procedure that the laboratory is following. If you have to measure potency has to be measured in every stage of the plant, and even sometimes when you are approaching to harvest, more times in order to have a clear view of the cannabinoids of your plant. It's very important to know before you cut your plants to go for drying what you expect to have. And also another question about microbiological analysis that we had. Unfortunately, in this day, we cannot cover all topics. Microbiological analysis is a very important topic, but at the moment, it's not included in this day, in this first Cannabis Excellence Day. In our second Cannabis Excellence Day, we will, of course, provide also microbiological analysis. I hope this is everything is clear now. Thank you very much, Tina. I don't see any other questions. We'll be glad that we have you here and provide us all this very useful information.
Thank you for having me.
Okay. So now we have a 5 minutes break before we move to the next presentation from company VTA. We will start sharp at 14:40 CET. Good evening again. Welcome back to the next session. Our next presenter is from company VTA, a German company that produces, among other, short bath distillation system for cannabis. Mr. Rafael Vargas-Sonner will present the VTA solution of the cannabis market. We welcome Rafael to our webinar, and he will inform us about cannabis hemp oil purification with short path evaporator systems. Rafael, you can continue.
Thank you very much. Hello, everyone. So I will share my screen. Yes, so welcome everyone. Thank you very much for joining the webinar about short path distillation for the purification of cannabis and hemp oil. My name is Rafael Vargas-Sonner and I'm a sales engineer at VTA. And we are working now with Hellamco for a little bit more than 2 years to supply short path evaporators for the growing cannabis and hemp market in Greece, Cyprus, and North Macedonia. First, I would like to introduce to you our company a little bit. So we are established in 1994. The company now consists of 190 employees, and we made a turnover of around about 50 million euros. We are a daughter company of the Stryker Group, which has manufacturing sites for mechanical devices and equipment in Germany and the Czech Republic. That's also where we manufacture the large short path and wide film evaporators. At our site, which you can see on the left, we also have a manufacturing hall where we manufacture the medium-sized and the small-sized equipment. And then in the high-rise hall at the end of left end of the picture, you can see our assembly hall. That's where we assemble all the parts of the industrial plants and ship them from there. And in the front, you can see the heart of our company, the testing center, where we develop the distillation processes together with our customers. We have here a great team which is doing trials on numerous different lab units, starting from feasibility trials with a small amount of material and product and up to pilot trials to scale up to an industrial plant. We make roughly 95% of our turnover with the supply of distillation plants, and 5% we make a turnover through the toll distillation. Toll distillation is a service where we take the product from the customer to our toll distillation plant here in the front of the picture, and we distill the product for them and send it back. We have 4 toll distillation plants. The smallest one has a throughput of 10 kilograms per hour, and the biggest one has a throughput of approximately 1,000 kilograms per hour. So customers which have to wait for a plant can use our toll distillation services. Now I have to check. I think I would like to show a video, and I think I didn't make the cross at the audio, so I check if you can hear the audio. 1 second. Otherwise, I have to...
Short path distillator is used for temperature-sensitive compounds and distills them in fine vacuum.
Yes, we can hear it.
You can hear it? Okay, perfect. Thank you. I wasn't sure if I made the cross at the sound. So I will show you a short video about short path distillation.
The product is fed in on the upper side and flows down the body by gravity. It is heated by heating media in a double jacket. The evaporated compound condenses on the internal condenser. The distillate and the residue flow out at the bottom. A wiping system distributes and agitates the product film for optimized evaporation.
Okay, so this was a 3D model of a short path evaporator, and I would like to explain to you now on a disassembled glass short path evaporator, again, the main components, which you saw for a very short time in the video. So one component is the evaporator shell. You can see here the glass body. It has a double jacket for the heating medium. So here, between this double wall is the heating medium. It can be either hot oil or steam. And that's the way that the evaporator is heated. Another main component is the here shown internal condenser. This is a spiral wound glass tube, and we pump through this condenser either water for temperatures between 5 and 120 degrees Celsius, or a glycol water mixture for lower temperatures. So the very special thing about the short path evaporator is that it has an internal condenser. So it's in the middle of the evaporator shell. And then the third main component is the wiper. Here you can see a black block. So these are the wiper elements made of graphite, and they are now disassembled, and when the plant is assembled, they are turning around the internal condenser, touching the inner wall. So the product comes from the inlet, flowing down through cavity, and it is then spread on the inner wall through this wiper. At the bottom, you can see the concentrate nozzle. This is basically a collection of the product which doesn't evaporate, which runs down on the evaporator wall and is then collected on the concentrate nozzle. The evaporated fraction, which goes to the internal condenser and runs down on the internal condenser, is collected at the distillate nozzle. And the name short path distillation, the name of the process is short path distillation, because you can see that the distance between the internal condenser and the evaporator shell is only a few centimeters, so a very short distance. And that is the reason why we have barely any pressure difference and pressure drop between the evaporator wall and the internal condenser. I will explain to you on the next one slide what advantage this has. To show this advantage to you, I would like to make a comparison between a lab rotary evaporator and a short path evaporator. Like this rotary evaporator. Here the product is in this flask, you can see here, which is in a hot oil or a hot water bath. So this is also an distillation apparatus, and the difference is that we have a higher and bigger volume of product in this flask, and it's heated. And after some time, some of the light boilers will evaporate. So this is a batch process, and the product stays at a higher temperature for a long time. So it takes several minutes up to an hour to evaporate all the light boilers. Then the vapors go to the external condenser. You can see here also marked, and they are condensed and run down to the distillate flask. And the apparatus is operated under vacuum conditions with this vacuum pump. Now, as I've already mentioned, one difference or one characteristic is that the duration, the residence time is very long, and another one is the pressure. So there are two reasons why the pressure is higher in a rotary evaporator than in a short path evaporator. One reason is that the vapors coming from the flask with the product have to pass through this small nozzle to get to the external condenser. And if you have a lot of vapors at a low pressure, then you will have here a very high pressure drop. Therefore, you will not be able to go below 1 millibar. And another reason is In this case, it's not so much because it's only a laboratory small scale rotary evaporator, but for large scale batch evaporators, you have a certain level of product in your vessel, and this and the product at the bottom of the vessel then has some hydraulic pressure. So you also have at the bottom a higher pressure, and therefore you cannot go down to low vacuum. Compared to that, the short pass evaporator is a continuously operated process. So it's a continuous process with a thin film of product. You can see the inlet, the product is fed in with a pump, and at the outlets, you can see the concentrate, gear pump, discharge pump, and the distillate discharge pump. So the time from the entry of the product to the evaporator until it is discharged is very short, around 1 minute, and it can be immediately cooled down afterwards. So the product is at a higher temperature only for a very short time. And the other reason, and another advantage advantage compared to a batch evaporator is that we have barely any pressure difference, so we can go down to 0.001 millibar. And then it's very important because you need a sophisticated vacuum system to protect your vacuum system. And we do this with our cold traps. Here you can see it. So any volatile substances that would go to the vacuum pump are frozen on the cold trap so that they don't contaminate the vacuum pumps. To show you the relationship between the pressure and the boiling temperature, I have chosen an example here, vitamin E. Here's the boiling temperature plotted at different pressures. So at a pressure of 1 millibar, vitamin E evaporates at a temperature of 210 degrees C. So that's already below the atmospheric boiling temperature. And with a short pass evaporator, we could go down to 0.01 millibar. So we could reduce the boiling temperature by roughly 100 Kelvin to 105 degrees C. So we don't need a high temperature to evaporate the vitamin E with a short pass evaporator. And it's similar for CBD and THC. You can see in the first line that the boiling temperature of these two substances at atmospheric pressure is around 410 degrees C. And at 1 millibar, it is already down to 290 degrees C, but this would be too high if we kept the CBD and THC for a long time at this temperature, we would harm it. Therefore, we use the short. That's the reason why a short pass evaporator is used, because it can go down to 0.001 millibar. And the boiling temperature is around 170 degrees, what is around 240 degrees or Kelvin lower than at atmospheric pressure. So, and thermal damage is a matter of time and temperature. And the short pass evaporator combines both, so a very short residence time and a significant reduction of the boiling temperature. At the end, we get a high quality product without any color change or polymerization if we do the short pass distillation correctly. To sum up our company and the introduction of the short pass distillation process, I would like to introduce you to some typical applications. So as I will explain later on, it's THC and CBD. Another one where we have supplied a lot of evaporators is the processing of edible oils and fats. For example, the removal of free fatty acids from vegetable oils. We have done this since the foundation of our company, and we have this is a large scale plant, so industrial plants. And here it's similar also with the removal of mineral oil contaminants like mosh and moor. We need very low vacuum conditions to reach minimum concentrations of the contaminants or of the unwanted substances. one of the main applications and our main business sectors is the concentration and the purification of omega-3 fatty acids like EPA and DHA from marine oils. We have also supplied here large-scale plants, and the purpose of the short-pass evaporator is to remove contaminants like pesticides to a minimum level. Yeah, so with at the minimum pressure of, let's say, down to 0.001 millibar, we are able to reduce at moderate temperatures the contaminants concentrations to a few ppm or even below. Another application is the processing of herb and natural extracts. We have supplied here mainly smaller and medium-sized plants. The customers have most times high requirements, so we have to build plants for GMP factories and with sometimes special cleaning procedures. So even though the plants are, let's say, a medium-sized or even small-sized, they are highly sophisticated. Another typical application is the purification of biodiesel and used oil. These two products contain some dark, high boiling components, and the customers, they want to get a clear final product. So we have to evaporate all the biodiesel and the oil in the short pass evaporator to get a clear, transparent product. And the last example I would like to show you is the removal of monomers from polyurethane, which is used as an adhesive or for coatings. And here we have to, we also need the very low pressure to reduce the monomer concentration so that it's compliant to the EU regulations. And all have in common that we developed the process in our testing center. So together with the customers, we did trials, first feasibility trials and pilot trials. And in the end, we gave them a process warranty consisting of a performance and of a quality warranty. So you can see here a package unit on the right side, and these are tailor-made plans which we design according to the customer's requirements. So the customer decides if he needs a control system, if we should build and design the piping, the pumps, the valves. And most of the times we assemble the unit together with the steel structure in our assembly hall. We have built evaporators with an area, heated surface area of up to 100 square meters. So these are huge evaporators with an inner diameter of up to 2.5 meters and a height of approximately 18 meters. So very large scale equipment, and our customers, we offer our customers pre-assembled plants. So the unit you see on the right side the picture was taken inside our assembly hall. So the unit was completely assembled in our assembly hall, and we did the vacuum and the pressure testing, and we disassembled it to modules with each a footprint of approximately 3 by 3 meters, and loaded it on trucks and shipped and delivered it to the customer site. And the benefit for the customer is that if we pre-assemble the units, then we install it in 1 or 2 weeks at the customer site, so there's no interruption of their production, or only very limited. So this was a general overview of the short pass distillation process and of our company. Now, I would like to speak about processing of cannabis and hemp. So before I start about the distillation, I will give you a brief overview about the extraction process, even though we don't supply this kind of equipment. So first of all, you have to extract the crude oil from the cannabis and hemp flowers. There are different extraction methods, as you will definitely hear also today in this webinar. like ethanol, cold or hot ethanol extraction, hydrocarbon extraction, or butane extraction. So different kind of solvents. And then, depending on the extraction method, you might have to do a dewaxing or winterization step to remove the waxes and the fats from the crude oil. In any case, you have to do decarboxylation to release the CO2. Now we are coming to speak about the distillation step, where we supply the equipment for. When you have the crude oil from the extraction, you have to run it twice over the short pass evaporator. So in the first pass, you will evaporate the light boilers, which are mostly terpenes. And the terpenes are a valuable product, so a high valuable product. You can use it for flavoring, and you can later on, when you get the purified cannabinoid fraction, you can blend it again and get a full spectrum product. Then the terpene-free crude oil has to be distilled a second time on the short pass evaporator because it still contains some high boiling components like waxes, lipids, chlorophyll, and they have to be separated from THC and CBD. In this step, we need a very low vacuum that the short pass evaporator can achieve, because we have to evaporate the cannabinoids, and then we get two fractions in the second pass. So the distillate is the final purified valuable product, and the concentrate, or here written residue, are the high boiling components like the waxes and lipids. So after the distillation step, you get two valuable fractions, the terpenes and the distillate, and one other, it's the high boiling components like the waxes and fats. And just to give you a complete overview, I also want to briefly mention that the cannabinoids can be further isolated, even though this is not our, we don't supply the equipment for this. There are processes like crystallization or chromatography. So in the last years, we have supplied, since 2016, we have supplied several lab and pilot units for the production of and processing of THC and CBD around the world. And we have developed two standard equipments. One is the VKL75 glass evaporator, you can see on the left. And it's our standard equipment. It's a complete unit. You just have to plug in the power cable, and then you can immediately start with the production. It is easy to operate. You operate it manually, and it's for a throughput of 1 to 1.5 liters of crude oil per hour. So that's very let's say helpful or very good equipment to start, especially if you have let's say not so large amount of crude oil. And then if you scale up your cannabis facility or hemp facility, we have the second standard equipment. It's a VK100 stainless steel evaporator. It's also a complete unit coming with all the devices you need to operate this unit. And even though the evaporator surface is only 2 times larger than of the glass evaporator, the throughput is significantly higher because this is a stainless steel evaporator with a better performance. So this is then for, let's say, medium-sized processing capacities. And if you want to further upscale your production, we will design for you a tailor-made plant for unlimited capacities, or for any capacity that you need. So I would also give you some background, some experiences that we made with different customers in the cannabis and hemp market. So since several years, we are working, and our partner, exclusive partner for cannabis and hemp in the United States is Root Sciences. They have a very broad knowledge about processing of cannabis, because they have sold hundreds of units in the United States, and they have made the experience that the processing is not regulated in the United States by the FDA. So they have sold mostly standard units. So as I've described before. And now in the past 2 or 3 years, we had one customer from Portugal, and they had the highest standards. They had very high requirements. They wanted to put the plant in a clean room. They asked us to build the plant according to the highest GMP standards. And so we had to polish all the product vetted surfaces. We made a control system for this small plant, and we programmed a step-by-step cleaning procedure. And even that plant was covered in a wall of sliding doors. Yeah. So, and what I would like to say about this very high requirements of the customer, they increased the price significantly, so that we ended up at a 5 times higher price than the standard equipment. But there's basically no difference at the output. So the feed rate is the same, and also the product quality of the purified material is the same of this plant on the right side compared to the plant on the left side. So if you want to build a GMP plant, then please, in your interest, build it to the minimum requirements of in terms of GMP, and don't set the highest requirements, even if they are not required, because then the price will increase significantly, and even though it's not necessary. Yeah? So it's good to spend here some extra time and to think what special requirements do we really need, and which are probably not required. And now, at the end of my presentation, I'm very happy that we had the chance and the help from our customer, Professor Skaltsonis, and his team from the University of Athens. They purchased a short pass evaporator VKL75, the glass evaporator, a little bit more than 1 year ago. And I'm happy to share his feedback here. So he says that they were able to distillate the hemp oil and purify it up to a concentration of around 87%. And then after some downstream process, they made this white powder of CBD. Professor Skaltsonis told us that they use this short pass evaporator for various purposes. For CBD purification, but also for the concentration of other substances like squalene from olive oil waste. And so there are basically no limits. This is really a lab and a R&D unit. You can use it for the development of new processes, and you can use it for small-scale productions as well. He said also that the unit, that he's happy with the support, especially of Hellamco. So that's a very big benefit, that Hellamco here is in direct contact with the customers, and that we are trying to help wherever we can. And he said that they are operating the unit now for 1 year successfully, so it's easy to operate. Yeah, at the end, thank you very much for your attention, and I would like to start the discussion now. Yeah? You can ask your questions.
Thank you very much, Rafael, for this very nice and useful presentation you had.
Thank you.
And now we are waiting for questions. If any questions appear, I will transfer it to you. In the meanwhile, as we are waiting for that, I would like also to express our customers' high appreciation and interest on the VTA equipment. We have already installed 3 sorbate distillators in Greece and North Macedonia. They all work perfect, and we are very happy about that. And we're looking forward for more.
Yeah. In a short time, we already had some great success. So I'm also very happy about that.
Yes. Since I don't see there are any questions, we will.
I can also share some.
You can share?
Yeah, I can share either a video or we have also made a case study about this the short pass evaporator of the University of Athens. I don't know if I can upload it, but
we still have 8 minutes, so you can share whatever you think is useful.
Yeah, then I will share our image video. So let me switch.
And guaranteeing successful production in various industrial sectors.
Wow. Could you see it? Yeah, okay, sorry. Let me try again.
Sector.
Let me try again. So because I stopped the sharing. Now I get it. I stopped the sharing. So I have to start again, and I will share it now, or is she sharing complete? So can you see it now?
Yes.
Can you see the
what
video?
Makes us?
Okay, then I try something else. Can you see it now?
Yes.
Okay, perfect. So I had it on the wrong screen.
Process engineering experts is not the sole ability to construct high-end distillation plants, but our knowledge and experience in finding excellent solutions, developing innovative processes, and guaranteeing successful production in various industrial sectors. A turnkey solution with process warranty.
This is what makes us experts in thin film and short path distillation.
It's our mission to serve your needs perfectly, with solutions as individual as your requirements are. From small scale to industrial size facilities. As an innovative company of the Stryker Group, we combine more than 25 years of experience with continuous innovation to bring together plant and apparatus manufacturing and customized processes. Everything starts with trials of your individual process in our laboratory to find the ideal solution for your requirements. With the possibility of toll distillation, we offer a customized production without the need of investment in your own production facilities. Our team plans and develops processes from the very first concept to the commissioning of your plant. involving mechanical, process, and electrical engineering capabilities. For you, it's only one contact person. In-house manufacturing of key process equipment ensures highest quality standards, designing, assembling, and shipping plants as skid-mounted package units. This is our profession. This is what we stand for. We are VTA.
Okay.
Okay. So if you want, you can share also the case study, to see the case study prepared for the University of Athens.
Yeah, exactly. Is it possible to send it to the customer so everyone can download it, or should I just share it?
Just share it.
Okay.
At the moment. And then it's available, so every customer that needs that can ask for that to be delivered to him.
Yeah, exactly. So if anyone would like to get a copy, then we can. Yeah, he can contact you, and then you have it. So.
Exactly.
So I think you can see my screen.
Mm-hmm.
Yes, so that's the case study about Professor Skaltsonis and his team from the University of Athens, which I already described briefly on this last slide of my presentation already. So yeah, here that's the VKL 75. And here we are describing that in 2018, the Greek government legalized the processing and production of cannabis and hemp for medical purposes. And we know that Professor Skaltsonis already had some experiences with short pass evaporator because he was working with it before. And he had... they needed some device to produce an end product suitable for their customers. So they reached out to Hellamco at that time, and that was basically the first project that we did together. So it's something like a pilot project, or something like a pilot project in Greece, and we're very happy that it worked. So that the customer has that the expectations are fulfilled, and that also the cooperation between Hellamco and VTA is also working so good. And so here you can see a picture of their unit, and here's a summary. So I showed you already this picture. Here are the codes that are already shared. So if you would like to have a summary of this case, of basically this presentation also, because it's like a small summary also with our contact data here from Yannis, who we are always working together if we have a project coming up. And also our website, and so I'm happy to share it, and we will also publish it online, so it's available.
Okay, Rafael, thank you very much for all your useful information. Since there are no questions I see from the attendees, we thank you very much again, and we will move to the next session.
Okay. Thank you too, Yannis, for the invitation.
Thank you very much. Have a nice day.
Have a nice day. Bye.
Bye. So it's time now to move to our next presentation. The next presenter is Nicholas Xinos from company THAR. THAR is a company in Pittsburgh, USA, producing SFE, supercritical fuel extraction, and SFC equipment, supercritical fluid chromatography. Nicholas Xinos is the global R&D and EMEA sales engineer, the technical expert, and he will present us the solution from THAR particle purification in efficient, sustainable, and compliant technologies for the commercial scale production of pharmaceutical grade cannabinoids. So, Nicholas, you can go on.
Hello, everybody. Thanks for being here with us today. So I'm Dr. Nikos Zainos of THAR Process, and we are going to be discussing today about efficient, sustainable, and compliant technologies for the commercial scale production of pharmaceutical cannabinoids. So THAR Process history goes back to 1990. Its foundations was built over 3 decades ago when THAR Technologies was founded. Then various spin-offs were created and then were annexed from THAR Process. Finally, THAR Process developed a CGMP pharma-grade CO2 extraction equipment facility, and in 2019, it manufactured probably the largest supercritical CO2 extraction system in the world, a 4-ton per day CO2 extraction system, together in 2020 with the world's largest supercritical fluid chromatography purification technology that was designed and dedicated for cannabinoids. So, first of all, we should ask the question why one should consider supercritical CO2 as a solvent. Supercritical CO2 has high extraction efficiency, and the systems that are being designed by THAR Process today are using CO2 as the main mobile phase, which has many, many beneficial properties. So that practically means that you have either no toxic solvents used or very small amounts of no toxic solvents, such as ethanol, which can be bioethanol. Then superior solubility over traditional solvents for both extraction and chromatography is what characterizes CO2, which is then leads to, in turn, to cost-effective solution from extraction to compound isolation. So that allows easy recoveries, much lower organic solvent consumptions, lower disposal of solvents, and this is very heavy for large scale extraction facilities. Less steps in standard operating procedures and post-purification endeavor, and very important for the chromatography and purification, much longer stationary phase life cycles. So overall, supercritical CO2 technologies are green technologies for sustainable growth, and supercritical CO2 technologies, they fulfill the environmental, social, and governance criteria. So that's why users of this technology fulfill this criteria and can apply for ESG funding, etc. So CO2 is cleaner, cheaper, and safer. It's non-toxic, it's a green solvent, it is stable, it is non-flammable. Actually, it's a fire suppressant, so it does not require X-proof facilities. CO2 itself is reclaimed from industrial plants. So basically what we do is that we are upcycling upcycling CO2 and then using it as a supercritical fluid for extraction chromatography. It's a fast technology that promises high throughputs. CO2 is recyclable throughout the process of both extraction and chromatography, and this minimizes wastes. And it is a sustainable technology, especially as far as the heat exchangers are concerned. So this minimizes energy use. So supercritical CO2 extraction and chromatography were invented long ago, and some segments of the natural product industry use these technologies, as their favorites. It was not invented for cannabinoids. Cannabinoids was a perfect fit when cannabis started legalizing. But way before that, it was utilized for fish oil and algae oil omega-3 fatty acids extraction and purification, DHA and EPA, for pharmaceutical small molecules, APIs, or enantiomer separation for pharmaceuticals, antioxidant, polyphenols, flavonoids, etc., such compounds, terpenophenolics extraction and purification, natural products, biomarker extraction and purification overall, and of course cannabis. And then what came into the game right after cannabis was the fact that supercritical CO2 proved to be an ideal solvent for synthetic lipids purification. These lipids are today being used in the mRNA vaccine manufacturing industry. Excuse me. So what is a supercritical fluid? Supercritical is a state of the matter where no distinct liquid or gas phase exists. This means that by increasing temperature and pressure for a certain molecule, we can reach its critical point, and above this critical point, in terms of, again, of pressure and temperature, this molecule is in supercritical state, and it is called a supercritical fluid. This critical temperature and pressure for CO2 are relatively low. Temperature is 31.1 Celsius and pressure 73.8 bar. So above these conditions, we have supercritical CO2 rather than liquid CO2. This means that CO2 in this state, it has a very high diffusivity comparable to this of a gas, but at the same time, it has a very high solvating power comparable to this of a liquid. So it combines the optimal properties of both gas and liquid for extraction. We can control the extraction and chromatography parameters. These are tunable, and this is critical to optimize and achieve reproducibility of our processes. So we can fine-tune polarity and selectivity by fine-tuning pressure and temperature with supercritical CO2. And doing so, we can focus and target on various different natural products and categories of classes of compounds that will be targeted from within the biomass, the plant matrix that we are being processing. So the key parameters that are being explored for process control and optimization are basically four: pressure, temperature, co-solvent percentage, whether we can use ethanol and at which amount, and flow rate, which will in turn lead us to a specific solvent to feed ratio that will be optimal for each application. So when we compare supercritical CO2 systems, we need to always have in mind that it's not the volume that counts, is the solvent to feed ratio. So it's basically the solvent to feed ratio that will define daily throughput. And this heavily depends from the pump capacity and the flow rate that this can achieve. This in turn heavily depends on the existence or not, for poorer systems, of a Coriolis mass flow meter. As far as sample preparation is concerned, it's the particle size and the moisture content of the biomass that are important. And then, of course, it's for large-scale, commercial-scale systems, what's critical for reproducibility is the possibility to run unattended and to have a compliant control software in order to reduce the variability of humans. So, THAR process product portfolio for cannabis facilities includes two main equipment classes: extraction and purification. THAR process manufactures both based on supercritical CO2 technology, and both two are fully EU GMP compliant. So THAR process systems will come with GMP packages that are available from THAR and will include and satisfy all requirements that will be needed for the auditing from national medicines agencies, local authorities. THAR process has two robust revenue streams. It is an equipment manufacturer, principally, but at the same time, THAR process is an R&D center and toll processing center for applications that will involve its own manufactured equipment. This means that in Pittsburgh GMP facility, we can process up to 4 metric tons of biomass per day. And this is done by utilizing the very same systems that we are manufacturing and selling to the market of extractions and pharmaceutical market. So focusing now into cannabis. Phytocannabinoids are contained in cannabis-derived extracts, but these extracts will contain probably more than 500 identified secondary metabolites. Phytocannabinoids alone are more than 100 compounds, different compounds, and terpenes might be more than 140 different terpenes. And then there are other classes of compounds like flavonoids, phenolics, sterols, of course, chlorophylls, etc. So, but the phytocannabinoids, which are our target for both extraction and purification, chemically speaking, are terpenophenolics, which means that they have a medium to relatively low polarity, that is a hydrophobic nature, which in turn depends on whether we are talking of the acid or neutral form. Because, as probably most people already know today, the phytocannabinoids are mostly present in their acid form in the trichomes of the cannabis flower, but it is the neutral form that is being utilized as bioactive compound in medicinal health and food products. A workflow from plant biomass to material ready to extract and then down to even isolated compounds will include various steps that are summarized here. Cannabis is harvested, then various post-harvesting steps are performed like de-leafing, buckering, and trimming. The material will be dried down to moisture below 15 or even better 10%. It will be ground to a particle size that will be below 1 millimeter. Then it will be extracted with our THAR process super fast supercritical CO2 equipment. Winterization, filtration, and decarboxylation, of course, then short path distillation, and then again with THAR process purification system, individual cannabinoids will be purified from the short path distillate. So, as discussed before, the two crucial steps in this process flow diagram, extraction and purification, will be undertaken within our facility and hopefully within yours by utilizing THAR process supercritical CO2 extraction and purification systems respectively. First, discussing extraction, THAR process has 6 different systems available. Those are standard systems, starting with a 2x9 liter, 2x25, 2x120, 2x360, 2x720, and 2x1650 liter systems. So with these systems, we're able to process somewhere between 15 kilograms to 3 or even 4 tons of biomass per day, depending on the biomass, on the level of the grinding, so the bulk density, etc. All these systems are equipped with recyclers to economize on CO2 consumption. And our process has optimized its systems for cannabinoids by designing a specific terpene collector that will have the task to collect mainly the mono and sesquiterpenes of the cannabis in a step that will be a step prior to cannabinoid extraction, but introduced into the same batch program. So this is automatically done by our systems, and the terpenes will collect in a separate extractor. So what will be collected in separator or collector 3 will be a liquid form of the terpenes, while in separators 1 and 2, we will have the cannabinoids and waxes separately. This means that supercritical CO2, with its fine-tunable parameters, can act as various solvents. It means it can sequentially extract and fractionate from the same biomass matrix different classes of compounds. And this is what also greatly differentiates this solvent from ethanol, hexane, so other organic solvents. So the extraction efficiency is being measured in our in-house analytical lab with robust analytics. We have HPLC systems, UPLC systems, GC systems. So we have our own analytical lab with validated methods. We even perform third-party analysis for other players in the cannabis industry. And so we are in position to fully determine structure. So it means we can fully determine the quality of our extracts, and then we can quantify the compounds, the extracted or then even isolated compounds from our extracts and fractions. This is how we have measured that we can go beyond 97% or up to 100% in recovery of certain cannabinoids. But what counts when one has to make its decision about whether doing supercritical CO2 extraction or ethanol extraction or any other kind of extraction. But of course, it usually compares to ethanol extraction as far as cannabis is concerned. What counts is basically not the capex of the extraction system alone, but the so-called total cost of ownership. And this is what we have been able to measure for both a supercritical CO2 extraction platform equipped with THAR process system in comparison to an ethanol extraction, cryoethanol extraction platform equipped with European-made cryoethanol extraction system. So by doing this sort of comparison, apple-to-apple comparison, we have been able to estimate the total cost of ownership, including not just the capex of the system that will do the extraction, but also, first of all, the capex of any auxiliary system that comes together. It means when you're using ethanol or any organic solvent, you have to have equally dimensioned solvent rectification systems in your facility. Then you have to calculate solvent disposal costs and then new solvent coming in all the time, while CO2 is being recycled continuously throughout the supercritical extraction process. You also need to calculate, of course, manpower for the extra steps, like the solvent rectification and energy usage for these very much energy-consuming extra steps that involve higher temperatures, and reduced pressures. So overall, this will lead to a very beneficial supercritical CO2 extraction system platform, I would say, total cost of ownership. Now, discussing purification with supercritical CO2 chromatography. I/C is like HPLC, like a high-pressure liquid chromatography in process scale level, except that the primary mobile phase is supercritical CO2 instead of an aqueous organic solution. So it's an alternative to LC, to HPLC, or even flash chromatography, but it is more sustainable. It's a green technology that allows for greener processes and greener outcomes, greener products with less residual contents of organic solvents. So CO2 is way less polar than water, right? So it's similar, its polarity is similar to heptane. And together with CO2, we will be using very small amounts of organic solvent. But in the case of cannabinoids, this will be ethanol. So we are typically using between 2 and 3% ethanol for cannabinoid purification with supercritical CO2. This means that the rest is CO2. In contrast to any liquid chromatography where you have 100% organic solvents or mixture of water with organic solvents. So, and these two have drawbacks. Water has the drawback that it's difficult to remove it from the fraction due to its very high boiling point, and organic solvents are toxic. So it's easier to remove, but they are toxic, they need disposal, and they have a certain cost, which is higher than CO2, by the way. So another advantage is that the flow rates of supercritical CO2 chromatography are typically higher in comparison to LC. So let's say for the same footprint of equipment, you have higher throughputs. And then Of course, to do this, you need to have a back pressure regulator in your system. But then this is probably the major difference from an LC system at process scale. So CO2 offers less solvent consumption, less solvent and stationary phase disposal, and lower overall operational cost and environmental impact. Our fractions are collected in small volumes of solvent, and this is a volatile solvent such as ethanol, which allows for very easy and fast recovery. Essentially, you have a very small rotary evaporator compared to what you would have with any LC, HPLC, or flash chromatography system. It's faster, so this leads to higher efficiencies and throughputs. CO2 is continuously and easily recycled throughout the process without bearing any high energy costs. And SFC applies to many small pharmaceutical molecule separation, including cannabinoids. Overall, it has a profitable TCO, total cost of ownership, compared to alternative technologies. THAR process isolator series include 3 different systems. The SFC10X that can purify up to 3 kilograms per day of material per day. Isolator SFC22X that can purify up to 20 kilograms of material per day. And then isolator SFC60 that will purify up to 65 kg of purified input material per day. So THAR process also has its proprietary series of purification media, stationary phases, and we'll supply this to the customers. And these are the stationary phase with which we have optimized our processes to be as fast as possible in terms of chromatography and to consume as low ethanol as possible. That's how we basically remain between 2 and 3%. And all the systems are equipped with recyclers in order to economize again in CO2 consumption. So we can purify any individual cannabinoid, CBD, Delta-9 THC. We are working a lot with the hemp industry for Delta-9 THC remediation. We're doing minor cannabinoid purification, and we have equipped the major minor cannabinoid manufacturers and suppliers in Europe, together with other pharmaceutical companies in the domain. We're doing terpene purification and even pesticide remediation through chromatography. But one needs to understand that this technology is not dedicated and unique to cannabinoids. It's a technology that's also being used for major other applications like synthetic lipid purification used in COVID vaccines, right? So today we have supplied our isolator systems to the major COVID mRNA lipid manufacturers for their synthetic or sometimes natural lipid that they need to purify, which is the main excipient for the mRNA vaccines. So here's an example of THC remediation from our facilities in Pittsburgh. We're separating the CBD from the THC and the other cannabinoids. Here's how we stack injections in order then, after we have developed the method, then we want to maximize throughput. So we have stacked the injections to maximize throughput throughout the day, of course, running unattended through the night, as long as we make sure we have enough solvent and enough material to inject, and of course, enough CO2. Here is real-world examples of THC remediation fraction analysis with concentrations of CBD and THC. So we can go as low as to non-detect THC in this case. And the same applies, so we can go more than 95, 97% purities of APIs, cannabinoid APIs. This is our method, basically. So we, and of course, so we supply the method together with the system, and then we support the customer for integrating this method into standard operating procedures for their facilities, their pharmaceutical facilities. So we give basically right away the data and the info for you to run the extraction and the chromatography as it should be and as it is optimized within our R&D and toll processing facility. So you see ethanol is only a very, very small portion of what we're using in flow rate for CO2. 125 bar pressure, 15-minute cycle. We are injecting every 15 minutes. Very fast cycle of injection. So this way we can achieve a 2.2 kg throughput per day. And then focusing on minor cannabinoids, where we start very, very low in concentrations. Here's an example of how we purified cannabinoid from initially starting from somewhere around 3% to first around 45%, and then second run 84%. And then again, here's how we're stacking our injection through the usage of our 21 CFR Part 11 compliant software system. This is how we do compliant processes, but also efficient processes, and with compliant equipment. Total cost of ownership counts everywhere, so it also counts for chromatography, and this is a rather complex thing to calculate and to do in order to apple compare various chromatography systems, because one needs to understand the processes and have real world data in order then, and from, of course, for a long time of operations, in order to be able to produce such a comparison. So our experts have this expertise, and that goes beyond supercritical CO2. We're working with partners, with customers, with scientific experts, and we have performed these comparisons that will show that utilizing supercritical CO2 will be very interesting in terms of total cost of ownership in, I would say, not the long run, but even in the short run. After a few months, the total cost of ownership will be equalized between any flash or HPLC system and a supercritical CO2 chromatography system. And this again comprises the solvent recovery auxiliaries that one will need for each one of these platforms. You can imagine that one would need a way smaller solvent recovery system when utilizing mainly CO2 as a solvent for chromatography, in comparison to what they would need for a chromatography system that would utilize water and ethanol. That's the elephant in the room. And the second elephant in the room will be stationary phase life cycle. In flash or HPLC chromatography, the life cycle of the stationary phase will be somewhere between 50 and 500 injections life cycle, while with supercritical CO2 we have achieved more than 20,000 injections. It means that this very expensive consumable of the chromatography, that is somewhere between 2,000 and 8,000 euros per kilogram, will have a very different impact in your facility costs, in your OPEX, when you are using either supercritical CO2 or any other kind of chromatography. It will be very much lower when you're using supercritical CO2 chromatography. So, to summarize, our process product portfolio for cannabis facilities includes extraction systems and purification systems, right? So extraction from the ground and dried biomass, focusing on cannabinoids and terpenes, and recovering a solventless oil, and then purification for isolating individual cannabinoids or even THC removal for the hemp industry. And these systems are fully EU GMP compliant and come together with THAR's application knowledge in both lab scale and very large scale. Thank you for your attention today, and don't hesitate to address any questions in my email or through our estimated partner, Hellamco in Greece. Thank you very much again.
I'm afraid we will not have the time now to move to questions. In any case, you have any questions, you can send your questions to info@hellamco.gr and we will provide with the answers. In the next 5 minutes, we will be together with you with the presentation of CONVIRON. So give us 5 minutes to prepare and we'll be ready. Good evening again. We will now move to our next session. We will have a presentation from company CONVIRON. CONVIRON is a famous Canadian company manufacturing specific rooms and special rooms for cannabis cultivation. We have three presenters two presenters from CONVIRON and Mr. Hoffman, that is from the company Hoffman Consulting. So our first presenter is Sarves Ramlochun, that is account manager and for global production customers. Mr. Travis Sutcher, that is commercial sales manager for North America, and Mr. Michael Hoffman, director and owner of Hoffman Consulting, former vice president of cultivation of BZAM. So I pass now the presentation to Sarves, and we are waiting for the presentation.
Sounds good. Thanks, Yanis, and thank you to Hellamco in general for this opportunity to present here. I'll get started. Today's presentation is about CONVIRON's growing and drying chambers, and it's also about how you can improve your cultivation facility and basically use precision growing and drying to increase profits. I have an agenda here. So we'll start with introducing ourselves. We are CONVIRON and Argus. Next up, we'll showcase our offerings, which are grow rooms, drying and curing rooms. And then we'll have the perspective of Michael Hoffman, who is a CONVIRON and Argus super user. So about us, our company is CEL, Controlled Environments Limited group of companies. And under that umbrella, we've got CONVIRON and Argus. CONVIRON has over 55 years of experience in controlled environments for plant growth and plant-centric control systems. We are the world leaders in the research sector, and we're also really well known in the industrial sector. Argus Controls is a plant-centric environment controls and automation system. They're used in greenhouses and indoor facilities. They have over 35 years of experience in the sector. So together, we're able to provide quite a robust portfolio of offering for the cannabis industry. Our company is composed of 4 main offices for CONVIRON and 1 main office for Argus Controls. And we have a network of distributors, of which Hellamco Scientific is the main one for Greece. With that offering, we've been able to provide products in over 90 countries so far. And our main clients are either in the research sector, in ag bio, or commercial growing. Here's a short selection of some of our clients. Next up, to narrow down on the cannabis industry, as cannabis started being legalized in Canada, and our main office is in Canada, naturally, we used over our 50 years of experience in controlled environments to create a portfolio of products that are tailored for cannabis plant growth. And now we are in business with 8 of the top 10 in North America, and we're also expanding in Australia and Europe. So what do? We provide walk-in rooms and reach-in chambers for production, for plant growth and drying and curing. Those come in different sizes. Each application is designed to address key factors of plant growth. Some of them are airflow, lighting, humidity, CO2, and your growing demands themselves. So this is what our rooms look like in general. What we provide is an indoor growing environment. So that includes the box, that includes all the HVAC equipment inside. Everything you need to create that environment is provided by one company, which is CONVIRON. Now I wanted to dive a little bit more in our growth rooms just to showcase what they look like. We'll start with our clone and propagation chambers. So the clone itself is one of the most vulnerable stages, and that's because the plant is really delicate at this point. So high humidity and consistent temperatures are what's needed to prevent drying and allow for rooting. The airflow distribution is a gentle horizontal airflow. And as you'll see here, I've got a little parameter range for humidity, temperature, lighting, and airflow. As we go through different rooms, you'll see the same thing happen again. That's because we don't provide one set point. We don't just provide a room that can only reach one temperature. We provide a range, but once you select one temperature within that range, for example, we make sure that we're holding that with high precision. So you've got flexibility to try different things that works best for your cultivar, and we hold those environments constant. Next is our tissue culture room. So what's different about our tissue culture room compared to the industry in general is that we provide an air shelf upward airflow. What that does is it prevents condensation on the containers for your tissue cultures. Generally, because tissue cultures sit on a canopy of lights, those lights may heat up that container and cause air to rise inside. As it goes on top of the lid, it condenses due to the cold surface of the lid. With that upward airflow, we've reduced that temperature gradient, and we ensure that as air rises, there's no surface that's cold enough for it to condense. In short, what that means is the plants inside that container are receiving the same lighting environment. We're not creating any microclimates to increase moisture. And it's basically a uniform environment, but yet it's flexible as you can change the ranges, as I've mentioned before. Then we go to our veg rooms. Our vegetative rooms are the next step up from cloning. That's when your plants are now establishing a healthy root zone and they're getting ready to grow more. So the airflow is built to be a little bit more robust, to be able to match the transpiration rate that your plants will experience as they keep growing. With this, we've got a reduced relative humidity inside the room just because your plants are now transpiring more. So they need to be able to remove that moisture at a higher rate. We also have the system that will schedule lighting intensity. You can increase lighting intensity to match growth, and also you can decrease humidity as growth progresses. All of these little changes that you make, they are being saved in the system. They're all being managed, controlled, and data is being collected so that you can tailor your growing regimen. As time goes by, you can really try and optimize all those different circumstances to ensure that you have a recipe that's working the best for your strain. The next step up from this room is the flower room. With the flower room, we provide a downward airflow. So this is a more robust system because your plants are now releasing a lot more water because of the high transpiration rate. We match that with that bottom airflow. By doing so, you can finish up your final growth stage and increase your high light intensity, use longer day cycles to establish that desired plant mass that you want and ensure you have a good root base before blooming. And then you can decrease your lighting cycles and also decrease your temperature that will induce blooming. As you do so, you can tailor your environmental parameters to ensure that you're getting a really good resin production to increase CBD, THC, and all the cannabinoids that your plant is responding to. At the same time, because of the way the room is optimized for plant growth, you're preventing microclimates. You have reduced humidity, you have a good uniform airflow. So that means you have less chance of having pockets of environment where you can have botrytis, mold growth, yeast, all kinds of infection that plants could experience without a proper environment. Next is the mother room. Mother rooms are similar to what you've seen from the flower room section, but these are really established to maintain the plants in the veg stage so that you can keep on collecting your genetic information from those plants. And also a fun fact, this is Michael Hoffman in the picture here. Next up, we'll move to drying and curing. So this nice picture is provided by courtesy of one of our clients. Here, in this case, this is inside one of our rooms, and they're doing hang drying. They're hang drying branches. So that gives you an idea of what the room layout looks like inside. So let's go a little bit more on drying and curing. So drying and curing could likely be the most crucial aspect of production when it comes to creating high quality flower, because you can grow a great plant and have a great flower, but if you don't dry it properly, your customers will never experience what you were trying to build here. If you don't preserve those terpene levels, if you don't provide them with something that's cured really nicely, or it doesn't have the bud integrity that they're looking for, then the product is not necessarily going to move. That causes revenue loss, and ultimately you're not making profits. So what we do with our drying and curing rooms is it's a repeatable and consistent output room. Once you've set up your conditions, you've established how you want to dry, you can repeat that sequence over and over again. You'll get the same product at the end of it, and it'll be consistent throughout the room. That's because we have a uniform airflow, we have uniform environments inside, and the system itself, it's built and engineered based on your harvest capacities. We don't take a look at how much room you've got inside your drying area and just provide you dehumidification equipment for it. What we do is we take a look at what's your harvest capacity. We work with you and your facility to ensure that the harvest that you're trying to dry will be able to be dried properly inside that room. So it's engineered based on dehumidification that you need for your harvest. And one big thing about our rooms is because of our nature working in the research industry, working with ag bio and phytopharma, we've been able to design a room that mitigates microbial growth. The room itself is based on an engineering principle of removing 50% of the moisture in your wet product within 24 hours. By doing so, we're decreasing the water activity inside the buds to below 0.8. And what that means is there's less chances of microbial growth. In addition to that, our air intake have HEPA filters, which ensure that you're further mitigating any microbial addition into that room. And the room themselves, they are a positive pressure room. So once you close the door of the room, and you can see the picture of the door there, there's a slightly higher pressure inside the room than outside. And when you open the room, that means air will go out as opposed to air being introduced in. And with those little additions, it makes the room customizable to the point that we can achieve GMP compliance. Now, of course, we can't provide a GMP certified room because GMP certification is dependent on the equipment and all the processes around it. But what we can do is work with our clients to ensure that what they need to meet their EU GMP requirements, we're able to provide within that room. Furthermore, the room is flexible. As all of our rooms, you're able to really change your parameters and obtain that perfect drying curve that you want. And because you can dry at lower temperatures, it also helps with preserving terpene profiles. Because terpenes are volatile, the lower the temperature that you're using, the higher the chances are that you're really enhancing that amount of terpenes that being left at the end of the process. So next, I wanted to explain a little bit more on our drying curve. So there's a lot going on in this picture here, but I'll try to make it a little bit easier to explain. So the black curve here, that's moisture content inside the buds. This is provided by one of our clients. And they came into the room and measured the moisture content within their buds at regular time intervals. In green here is the amount of moisture inside the room. And you can see that it peaked here. That means all the product was into the room at this point here. They closed the door and they started the drying process. In orange here, you'll see that's the amount of moisture that's being removed. So that's the amount of condensate that the system is collecting at the end of it. So the system ramps up and starts drying. As it keeps drying and drying and drying, within about 24 hours, the moisture content within the bud has dropped to about 25%. So at the start, you were about 80%, and you've already dropped about 25%, which is more than the 50% of moisture requirement to mitigate microbial growth. So at this point in time, the customer who's drying notices that it might be drying a little bit too fast for their liking because they almost reached the desired 15% of moisture. So what they did is at first they were drying at 50% moisture, and they decided that they were going to increase the moisture in the room a little bit to slow down the drying process. So by having those optimizations and precision abilities inside the room, you're able to manipulate that environment a little bit to slow down the drying process and make sure that you're drying nicely, not over drying, not causing any loss in structure of your bud. So you can play around with this until you get something that you like. And because of this feature and increasing the humidity, you can also seal the room. So So you don't have fresh air coming in, you don't have exhaust air going out, so that you replicate a curing process. So you can use this room to cure as well when you're done drying. We typically say drying is about, in our rooms, it's about 3 to 4, maybe 5 days, and then the rest of the time can be used for curing. The good thing about this is as time goes by, you can perfect that drying curve, get something that you really like, and that's going to finally increase revenues. At the same time, it's repeatable, so you can use that over and over as time goes by. Here's a little bit of information of what you can expect when you come to CONVIRON. CONVIRON will provide you with your capacities, with everything that you need to ensure that performance will be reached by CONVIRON. And if we don't reach those performance values that we've said, you have a 2-year parts and labor warranty. So that means we will make sure that you're able to reach what you want to reach. We provide dehumidification rate. We've got standard rooms. We've got standard rooms with a preset amount of capacity that can be dried, but those rooms also modular. So we can expand them lengthwise to provide you with what you need. And the examples that I've shown have been with hang drying, but these rooms also do trim on trays. So if you were just drying flowers, also if you were drying whole plants, we can accommodate these as well. So to finish off with the CONVIRON product offering, I wanted to just summarize everything into the CONVIRON advantage. So what we are is we're accurate, uniform, and repeatable. We're a turnkey solution, so we provide you with the room that you want. We only need power to it. We only need connection to your chiller and boiler systems because we use your on-site utilities to ensure that it's more cost effective. But at the same time, you don't necessarily have to worry about sourcing all the different equipments inside it. We take care of that and provide you with the environment that you require. It's designed, installed, and serviced by one company, which would be CONVIRON and our distributor, Hellamco. We are a reliable solution, and it's been proven for all our experience in the research and commercial industry. We are built to last. We've got warranty. We offer local support. We are flexible to really dial in what you want out of your crop. And you can really use CONVIRON and Argus to ensure that you're getting an efficient process. So this concludes the CONVIRON offering portion. So now we'll let Michael Hoffman take it with an experienced CONVIRON and Argus user.
Thanks, Shavesh. Hi, guys. My name is Michael Hoffman. I've been involved in the medical and recreational cannabis markets now for 10 years. So that's a little bit about myself. And today I'd like to just talk a little bit about how to quantify high quality flower and just bring to light what is really what we should be defining as producing high quality flower. Just go to the next page, please. So defining quality flower, I think this is a really important thing because to me, I believe there's 5 criteria to really defining quality flower. The first one, of course, is THC and CBD, or the total quantified cannabinoids. The reason I say total quantified cannabinoids is because in a dry sample at 10 to 12%, typically what we would see is I've seen a range of total quantified cannabinoids from 180 milligrams per gram all the way up to 400 milligrams per gram. So yes, in the indoor world with the right strain, you can get an unbelievable high quantified cannabinoid concentration. Whereas if you're growing outside, you can get quite a low cannabinoid concentration. So it's quite interesting to see that the level of cannabinoids in a flower at 10 to 12% moisture can range quite a bit. The next one, of course, is the terpene profile and moisture content. So I believe moisture content, at least in a dried flower, should be between 10 and 12%. But once again, the total quantified terpenes. I've seen total quantified terpenes in samples between 1% and 5%. Once again, in an indoor climate-controlled room where we have control of absolutely everything, I've seen repeatedly results staying stable at the 4 to 5% range, whereas outside I've seen total quantified terpenes coming in at about 1%. The next one, which is a really important one, is any foreign content. The reason I say foreign content is mold, pathogens, and pesticides. So if it's a very dirty facility, you'll have very high mold and pathogens. Even if you irradiate and kill it, that mold will still be present in the finished flower, affecting the taste and affecting, obviously, the texture of the plant. Pesticides are another really important one, because a lot of growers don't realize we'll take a bottle of pesticide, we will constitute the pesticide with water to the necessary level we need to. So basically dilute the pesticide, apply it to the crop, and then we're going to stick it in the drying room and dehydrate all of that water, constituting that pesticide bottle back to its original form. And often there's testing standards don't catch every single one of those pesticides. So I feel in a high quality product, it's best to do the best we can to mitigate mold, pathogens, and pesticides. The fourth one I want to talk about is flower structure and density. This is more on the recreational side than the medicinal side, but also a good flower structure will hold more trichomes, and density will often have a higher concentration of cannabinoids if you get the density right. So flower structure and density can be an important part of growing a good quality product. And then the last one, of course, for growers is if we can achieve the first four, can we achieve that stable and repeatedly? And often that comes down to the stability of the genetics. So does that genetic have a repeatable high yielding capability? Go to the next slide, please. So how do we achieve high quality flower? Well, I think couple of things, starting off with like a CONVIRON solution, starting with a balanced design. And that's really important. So understanding basically your hydronics and understanding the cause and effect. So in order to achieve the standardized cannabis, every aspect of the facility does need to be understood, balanced, and validated. To understand the cause and effect of the equipment components, it's an important thing. What consequences can occur when equipment is not balanced and one piece of equipment is reacting against another piece of equipment? And ultimately, how will those plants grow in that environment? So understanding the biomass accumulation in the rooms, what happens when the plants start off small and move themselves to 1 meter tall plants? How will that affect the airflow modeling? How will that affect the heat generation? Another one is understanding light energy. When we increase light in the room, how is that going to affect temperature and humidity in the cultivation space? And of course, understanding irrigation events and how they will affect the humidity in the growth room. Move to the next slide. So I think it's really important to have control of all of those things. And the next important point to this is to have this all in one ecosystem. So to ensure all of the components of the production facility are in one operating system. This is ideal if we really want to produce medicine, because we want to be able to follow everything on one analytical data system from basically right in the mother room all the way through to the end of the drying room cycle. So if we can catch all of that on one data system, like the Argus system, we can do the climate control and monitoring. So not only applying the applications, but also catching the feedback. We can catch the irrigation processes. That's EC, pH, level of nutrients, precision of nutrients. And then, of course, also the drying and curing, and ensuring that there are no upsets in the drying and curing, and we're attending exactly what we're setting out to achieve. And most importantly, if we have this all in one data system, this will allow the software system to automatically measure the cause and effect and calculate the deviations. And yes, the Argus and CONVIRON solution has these capabilities tied into the system, which is really important, because when we want to continue to optimize our growth and look closer, we're constantly scouring for the deviations to look at how far off we deviated from what we intended. Ask the question why we deviated, figure out a way to mitigate that, and move on and build a building block this way. This is a really important feature of developing quality cannabis. Next slide, please. So design considerations for biosecurity is another one. We can build the perfect mousetrap, but can we keep it clean? So a couple of things I like to look at is obviously the two-door system, one door for the product in, one door for the waste out, one door for the cleaning crews in and out. And then of course, once it's cleaned and sealed and sanitized, the back door can be closed and the front door can be opened, and you've got a clean room to start your cultivation practices. So starting with the design components at the facility level to prevent cross-contamination can really be beneficial. One of the other things, of course, is ensuring the design components to facilitate the cleaning of the irrigation lines and prevent the buildup of biofilm. So how do we design that? And is the cultivation space designed to protect the crops and prevent cross-contamination from one batch to the other? So once again, if we want to mitigate pests or obviously really hamper pests from moving around, growing crops in single rooms, one batch, one room, will very drastically mitigate some of those things. You can move to the next slide. So design considerations around preservation of product. Well, of course, to understand preservation of product, I guess we have to ask the question, what does cause the degradation of cannabinoids? And what does cause the degradation of terpenes? To basically answer that question will, first of all, help us to mitigate it. So to truly understand the volatility of cannabis and what causes those degradations is an important step in creating a repeatable high-quality cannabis. Go to the next slide. So I think an important part of it is selected genetics. You can build the most beautiful facility in the world and you can think of all of those things. I still believe if you do not have the right genetic that is conducive to the environment that you're intending to grow in, you're not going to be there. To what I've learned so far, I feel it's about 60 or 70%. The strength of the genetic will overcome a large portion of this. So, of course, the question we ask, where are we growing? Indoor, outdoor? And we're looking for a genetic, obviously, that has the highest medicinal compounds. We're looking for a selected morphology of a plant that will fit to the cultivation practices, i.e. in the indoor world, we're looking for something that remains small, that produces a predominant high quality flower at the top, has a very apical dominance, does not shoot a lot of lateral shoots. In the outdoor world, we're looking at something that would be more self-supporting, more disease and pathogen resistant, early flowering times, depending on season or area that you're cultivating in. So basically really selecting that genetic to make sure that you're putting your best foot forward. Natural lower growing leaf indexes is important for the indoor world. Obviously genetic vigor, flower time, yield, and then pest and pathogen resistance is also a very important part of it. I think once you've found a really good genetic, after the last 5 years now, I've become a huge fan of working with tissue culture. I've learned that the amount of diseases that are being passed around in cannabis strains is unbelievable. And the amount of cleanup work we've done to a large amount of the genetics and what we have inadvertently found in the genetics once we've started tissue culture programs is mind-boggling. So once you've found a genetic that's really worth cultivating, I believe that resetting the plants with a tissue culture program will become vital in the next stages of this evolution. And starting with stage one cuts, we're propagating pure cells of the desired plant and moving that to a stage 4 and ultimately bringing that into the production cycle. Starting this way will drastically mitigate pests and pathogens and also keep the foreign content down in the plant. And will keep your facility clean. So developing and partnering with a tissue culture program is a very important step in consistent quality cannabis. So once we've got that, the next thing, of course, is the training and documentation. Is another important step, is to have a very clearly defined cultivation program that's easy to read, easy to follow, and to have proper training that can be followed to the desired cultivation program will, of course, increase the chances of repeated successful results. So using program management software that can both track the defined growing parameters, as well as ensuring the results are documented. Doing those two things, we can look and we can analyze the results and determine the level of deviation from batch to batch. And so once we've achieved that, we've got a standard growing practice. I believe validation is ultimately the last step in being able to repeat the repeated results. So once all of these things are done, I like to track not just one crop, but I like to track a series of as many crops as possible. So one crop after the other, and keeping very detailed notes of how we are moving along. So some of the important information to follow, I believe, is obviously the batch and lot number, the date and planting date, the strain. It's good to have an expected yield and an actual yield. If we know exactly what we're expecting and we're honing in closer and closer and closer to our expected and actuals, this is just providing the finance team and providing the people at the top a much better runway of where we're actually going. Flower to trim ratio is another important one. Looking at the flower to trim ratio is a very important aspect of how well a cultivator is doing. If we look at a 65-35 split, high quality flower, 65-35 being the trim, that's going to indicate that the grower is really producing a high quality flower. Inadvertently, if you have an 80-20 split where there's 80% trim that's coming off the flower and only a 20% retention of flower, it's going to show a real red flag as to the fact that the growers are not producing what they're intending to produce. Of course, tracking the THC, CBD, total quantified cannabinoids, batch after batch after batch, is also a very important part. Are we staying close to where we're wanting to retain our medicinal compounds for that product? Maintaining that 10 to 12% moisture, and of course, tracking the total quantified cannabinoids or total quantified terpenes. Once again, an important thing. If we produce a batch that's at 3.5% to 4%, can we repeatedly repeat that? Can we also repeatedly repeat the 12% moisture? Well, of course, with a CONVIRON drying room, you absolutely can do that. Following just basically the hydronics of the systems, like Sylvester pointed out, it really is the science of just being really key on those tools and making sure that you can repeat that result. Of course, number of plants, it's going to impact shading, impact a bunch of things. So once again, we want to see the number of plants always staying stable in our validation process. The next two are also important. The grams per square meter. Tracking this will show us, of course, how we're doing on the overall grow, and of course, how the financial projections are going to look. But not just the grams per square meter, also the grams per square meter per day, indicating how many days the plant has been in the room and indicating on a per day basis how many grams we're increasing with those flowers. And then, of course, the last thing we want to make sure is that there is any allocated R&D production, that we're measuring that as well, so we can minus that off the totals. So only after measuring all of these things and keeping an eye on the historical crops, I believe this is where we can get as close as possible to the validation of truly repeated high quality cannabis. So that's it for me. Thank you.
So this completes our presentation, but we are open for any questions that you may have. Our panelists for the question section would be myself, Michael, and Travis.
Okay, thank you very much, three of you, for all this useful information. We have a couple of questions already from the audience. One question is, what's the difference to yields if we compare a greenhouse cultivation system, greenhouse cultivation scenario, and the indoor system that's provided from CONVIRON?
Quite a difference in yield was the question?
Yes, what's the difference in overall yields?
So this is an interesting question, actually. Difference in yields, I think, as the industry continues to move forward. If we want to talk about yield versus quality, first of all, in Canada, we have a saturated market where there is more than enough low-quality cannabis in the space. People that are growing outside in 200-acre farms these days can produce a copious amount of low-quality cannabis. I believe what's happening now is we're seeing a shift in the industry where there is a lot of low-quality cannabis, but the one thing that's failing to meet the criteria is the high-quality cannabis. In Canada, there's a very low amount of high-quality cannabis that's meeting the specifications that we're looking for with the high terpenes and the high total quantified cannabinoids that are in the product. So once again, yield is not going to become, I think, the future. I believe if you're wanting high yield, you can grow it outside and grow it in boatloads. But it's going to either go to an extraction or you're going to have a very difficult time selling it. The indoor world will always stay where we're looking for the highest quantified cannabinoids, highest quantified terpenes, the flower structure, and we're trying to really hit all of those things. And the price will stay stable and the price will reflect as it has in Canada over the last couple of years.
Okay, thank you very much, Michael. Also, there is a second question to the tables that Sarvesh provided at the beginning of the presentation. You mentioned that there is different light demands for each stage of the growing, different light demands for clones, for mothers, for vegetation, for flowering. How can this light be measured inside the room?
Yeah, so to measure lighting inside the room, the easiest way of doing that is having sensors. At the canopy level, you can have sensors that are measuring how much light is shining. Those sensors will relay back to the system, and you can really input the microvolts that you want of lighting so that the system adjusts itself and gets you where you want it to get you. So the general demand is, as growing goes on, you need higher lighting intensity, and that's because you want to promote photosynthesis. But it's not the only thing. There's also the spectrum. So the lighting has different wavebands, different spectrums inside it, and you want to really tailor that to your cultivar and tailor that to your stage of growth.
If I can jump in on that one, Sarvesh, one of the, I guess, features in addition with Argus being tied in with the CONVIRON solution is that when you have those differences, like say you're changing a lighting schedule, you can actually have the system programmed to be preemptive for that scaling. So you're not getting that hard stop or differentiation in the lighting. Is you can control the HVAC or your airflow based upon that expected change to come so that the ramp up is steady rather than getting a hard start and stop based upon different environmental factors.
Yeah, exactly. And to go on a little bit more, what we're trying to do with these rooms is we're replicating an outside environment, but making sure that it's precise and constant. So as Travis just said, we want to ramp up. So we're going to replicate sunset and sunrise in a way, but make sure that we're keeping stable and consistent environments when we do that. Go ahead, Michael. I think I cut you off.
No, it's okay. I was just saying that's a really good point, Travis. And definitely with the replication of ramping it up and ramping it down is also helpful on the hydronics of the systems.
Okay, so I think there are no more questions at the moment.
If there's no more questions, Giannis, one more thing I just wanted to throw out there. As Sarvesh mentioned early on in the presentation, we are local to Canada. Obviously, with our global presence, we have no problem supporting areas of Europe, in particular Greece. I wanted to mention that our rooms themselves are fully CE certified, and you're basically dealing with a reputable organization that's been in business for over almost 60 years now. And is defined for precision growing environments. And in summary, basically, you can take those precision environments and tie that directly back to profits and yield. Like Michael had said, is if the focus is on yield, you can get low quality, likely in abundance in a short period of time, but it's going to be the high quality that really differentiates a grower and the longevity of that brand and their product.
Yeah. And also looking at the total cannabinoids or quantified cannabinoids and terpenes in the product now. Canada, we're really shifting now to quality indicators where what is in those cannabinoids, what are the concentrations of those cannabinoids, what cannabinoids are spiking in each strain, and then of course that's also to the terpenes as well. There's a big difference between 1% and 5%. And like I say, I've seen 1% repeatedly in the outdoor strains, outdoor grows, often sometimes greenhouses. And I'm increasingly seeing 4 to 5% now in the indoor world. And of course, it's just understanding the volatility of cannabinoids and terpenes and how we can change the hydronics of the systems to preserve those. And at least in Canada, that is becoming where the rage is there's a lot of low quality product out there, but the high quality product is still commanding a very good price and is in very high demand because it's very difficult to do.
Okay. Thank you very much, all of you, for your participations. It was really, you shared with us very valuable and very useful information. Now we will have a 2 minutes break, and then we will move to the next presenter, the last one, from MOBIUS. And Amanda James just informed me that they had an issue with sudden bad weather in the East Coast, in the West Coast in the USA, but I see she's connected. We'll arrange the presentation and we'll speak in 5 minutes. Good evening again. We are ready to start our next presentation. The presenter is Ms. Amanda James. She's from company MOBIUS in the States, and she's Director for Strategy and Business Development. Unfortunately, there is a weather issue there that Amanda James is located. There is no power over there, so we have to do the presentation. I will share it with you through my PC, and Amanda will just prepare to do the speaking. Okay, Amanda, you can start.
Thanks very much. Yeah, my apologies to everybody in advance here. We're having a massive windstorm, and at 7:43 in the mid, just at the tail end of the last presentation there, all the power here went out. So we're scrambling a little bit. So thank you very much for dealing with the presentation side of things. There are a few updates to the slide deck, and I assume that the slide decks are being made available to participants after the event. Is that correct?
Yes, this is correct.
Perfect. Okay, I will ensure that the more up-to-date version of the slide deck is what is shared with participants after this, and we'll do our best to get through this. So if you can move on to the agenda on the next slide. So today I'm going to be talking about a lot of stuff that if you're already in operations, it might seem like it's sort of, oh yeah, I've already got that. But there are always some things that we can do to really dial in our efficiency and our workflows. So I'm going to be starting with topics around that. So how to lay out the your processing area and how to get material in and out of the space, how to staff your processing area in a way that's going to work most efficiently. And then we're going to spend quite a bit of time on cleaning and GMP. And that's where I've got a number of updated slides that'll be shared afterwards. And then at the very end, I'll just do a really, really quick overview of the MOBIUS lineup of processing equipment. Okay, next slide. So this here is a photo of a tandem MOBIUS trimmer line, just to give you a sense of it. If we can go to the next slide. So when we're when we're setting up a processing room, if you are at the point right now where you are just speccing out your space, you have all the room in the world to be laying things out, but perhaps you're in a facility where you feel like you're locked in. I'd like to introduce some things here for you to consider if you have the ability to rearrange your space and get it working as efficiently as possible, or if you are in the process of planning, you can insert some of these things. So airflow is going to be critical, and it doesn't matter what type of equipment you're using or how much you're putting into your post-harvest room, meaning are you bucking in there? Are you trimming? Are you doing some packaging in there? You always want to make sure that you've got as much airflow as possible to manage the temperature. Your enemy, the moment that the cannabis plant is chopped down, your enemy is heat. So the lower you can keep your temperature, the longer you can prevent your plant material from composting on itself and from those terpenes beginning to pop. So managing the temperature in the room and getting some airflow is key. The last presenters had talked about this, so you want to be having, if you can, 2 different doors to the room. If you've got more, that's fine, but think of it like a high energy kitchen at a nice restaurant. You want people to be able to go in one door and out another. You want your cannabis material to be able to come in one door and out another, so that you're not having traffic jams and blockades on there. You can keep pressing. There are 7 items on this screen, so you can go all the way to number 7 there. You want to make sure that the room is cleared out so that you don't have any unnecessary items. Thank you. We don't want to have any unnecessary items in the space. so that we're not clogging anything up with other equipment. You want to have as much room as possible for your workflow. You want to take into account circulation space around your equipment so that you can not only get the product moving around efficiently, but make sure that you are operating safely. A cluttered, crowded space can lead to health and safety risks, tripping over power cords, potentially things spilling, so you've got water issues. So having that appropriate circulation space and the visual cleanliness of the space is really critical. Electrical connections, both for efficiency and safety, you want to have them as close as possible to the equipment. Do you need any other equipment in addition to your processing stuff? And that means things like scales or work tables where you would set up computers, anything like that. And then how close is your processing space in relation to where the equipment is going to be cleaned? If you have your wash bay on the complete opposite end of the facility from where your processing equipment is, you are going to be tracking your dirty equipment across your entire facility every time you want to clean it. So that is obviously not the ideal situation. So if you can, place that as close as possible to your processing space, because that's the equipment that's going to be need to be cleaned the most frequently. Next slide. And you can skip to the following one. So when we take a look at room setup, I'm going to be focusing a lot on trimming equipment over the next few slides here. And the reason why is because I find that everybody does bucking a little bit differently. Some people are hand bucking. Some people are using machines. Some people sort. Some people don't. Some people are wet or dry processing. But everybody is trimming. So I'll just talk about trimming specifically in this section here. So if you're using a trimmer that's set up with a vacuum style throughput, you want to make sure that the venting is towards the wall, not towards the center of the room, so you're not having everything fly everywhere. And try and take advantage of the shape of your room to maximize your efficiency. So the MOBIUS lineup of equipment, you can see in this photo, they have their QC conveyor, or sometimes we call it the outfeed conveyor. They have it set up perpendicular to the trimmers. Other places would have that set up in parallel. These people are just taking advantage of the shape of their room and making the most out of their space. Next slide. And there are a couple of bullets on this one. There are 4 bullets, so you can just show all 4 of those. Thank you. So the in-feed position, I call this the person who's on in-feed, I believe is the heartbeat of the room. And the reason I call them that is because they're setting the pace for everything else. If the in-feed person slows things down, everything else slows down in the room or becomes a bottleneck, and vice versa. So we need this person to be ergonomically comfortable. We need them to be set up for success, and so that they can keep things moving. That's what's going to hit your bottom line. So making sure they're comfortable with ear protection, anti-fatigue mat, good working height, making sure that they always have material on hand. So what do I mean by that? There are so many sites I've been to where they fill the in-feed conveyor up for the trimmer and they work through the last of the tote. And then that in-feed person has to go and search for more material to fill the trimmer up because it hasn't been brought to the in-feed side of the trimmer there. So there should be always a stock of material there for them, and they should be able to work really efficiently and swiftly, moving from one tote or one bag of material to the next. So again, it's back to having your space planning set up so you have enough room. Next slide. So in this little clip we've got in the background here, you can see the outfeed. And the outfeed conveyor is set up so that we can do a visual QC on the material. And this is not just for sort of the physical trim quality, but this is an opportunity for your people to be checking if there are any mold issues, things like that, that have maybe made it through to this point. So if we give people on the outfeed a small bin to put some material aside that either needs to be quarantined or touched up a little bit more than they can handle there on the QC, that's usually helpful. And again, we want these people to be as comfortable as possible. I always recommend that anybody who is sort of an operations manager or supervisor, either put yourself in the shoes of the people who are working the line, or work the line for a day and see how long you can concentrate for. Because if you find that it's very difficult for your people to carry out a task for any longer than sort of 10 or 15 minutes with the level of quality that you're looking for, you are not going to get the results. You're going to have lower throughputs and lower quality. So if you make your people comfortable and set up with the right tools, you're going to have the best chance for success at being really efficient and producing a high quality end product. Next slide. And maybe this slide is kind of talking about that summary there. So the in-feed conveyor on a trim system, whether it's a MOBIUS trim system or a different one, you want to be using that to get your consistent and highest efficiency throughput. So if you are hand feeding the machine without a conveyor, you tend to be pulsing the machines and you're not going to be getting a consistent throughput. That's going to impact your quality. This is an opportunity to catch any foreign objects before they go through the equipment. The number of times that I've been on site and been able to pull out a zip tie, an errant glove, even scissors before they go through the machine, because we're doing that visual inspection on the in-feed conveyor, is it happens. I would say at least 50% of the sites I've been to, the in-feed conveyor saves that from happening. It's also an opportunity to take out excess water leaves, and it's not that trimmers can't handle that, but it definitely dilutes the potency of your trim. So anything you can remove from the trim stream in advance is a good thing. The last presenters, still going through those points, Sarah. Yeah, thank you. The last presenters had talked about the quality of the material and the density. So this is a really, really big one. I cannot stress enough how, from a business perspective, how important it is to think about the strains that you are putting out to market. And rather than putting out 50 different strains, all of which trim up a little bit differently or dry differently, if you can focus on maybe 10 strains, a dozen strains, and they tend to have a morphology that has a high density, you are going to have a much easier time processing them, and you are going to essentially generate more revenue from them. You're going to have much more success there. When you're thinking about processing, if you are running larger batches, so not just sort of a 2 or 3 hour harvest, but let's say you're in perpetual harvest and you're processing 6, 8, 10 hours a day, you need to make sure that you've got what we call hot swap parts on hand, and that allows you to keep to do a very, very short break, swap out resin buildup on equipment for fresh parts, and keep moving and leave all the cleaning to the end of the day. So having that on hand. And then number 4 might be the most important one. I've been to a number of sites where they say to me, we're not hitting the throughput numbers that you say we should be able to hit. And then we observe the trim room, for example, and for every 60 minutes of their trim day, they only have the machine turned on for 25 or 30 minutes. All the rest of this time is spent finding totes of material because they ran out, or empty totes to put on the outfeed, or finding hot swap parts, or maybe they've gone for lunch or they had to break because one person was going on a washroom break. So The only way to achieve the throughputs that you're looking for is if you are maximizing the number of minutes out of every hour that your machinery is running. So to do this, you need to coordinate breaks. To do this, you need to make sure you have all the material in the room, that your in-feed never runs out, that you've always got spare empty totes on the outfeed, that you have a backup person to take over in case somebody needs to leave the room for a few minutes. So that's how you're going to achieve your maximum throughput. And related to that is the bullet number 5 there, keeping the trimmer full. If you're not keeping it full, you're not going to hit your efficiency targets. Okay, moving on to the next one here. And this is kind of the last one here around sort of the setup of your trim room. I always get asked, okay, how many people do I need? So if you are running a MOBIUS trim line, I recommend that you've got anywhere between kind of 4 and maybe 6 people on there. And I'll describe the different roles. So I talked about on the right-hand side of the screen there, you see the in-feed, and that person is setting the pace for the room. So that's a really important job. And I would recommend that when you staff that role, it's not the same person standing there all day, 8 hours feeding the machine. People only have an attention span for so long. So if you can switch the person up on that position, maybe every 30 to 60 minutes, I think that's an efficient way to go. The people on the left-hand side of the screen are on the out feed, so denoted by O, and this is very much a decision by your decision. Do you want any people on your outfeed doing QC? Do you want 10 people on your outfeed? Most places I go have between 2 and 4 people there, and what they're doing is just touching up, looking for crow's feet, looking for mold, things like that. And then the last person that we have labeled on this diagram, it says F for floater. So typically the floater is the most senior person in the room. So the floater's job is, or you could also call it a runner. If any of you have any experience in the restaurant industry, or you're like me and you just like to watch some of those cooking shows, this is also would be called the expediter. So if you're watching Gordon Ramsay always plays the expediter. He's yelling at people and getting them ready to go. So the expediter is making sure the in-feed never runs out of material. The out-feed never runs out of empty totes. Everybody's on task. The trim totes at the back, we're catching those before they get too full. We've got hot swap parts on hand and coordinating breaks and things like that. So your floater or your runner is keeping everybody in line. They're the general, making sure that everybody is carrying out their tasks. Okay, heading on to the next slide. So just before we move on here, this is a spot where in the in my revised deck, I actually inserted some links to some key GMP information. We have a GMP guide on the MOBIUS trimmer website. You'll have that link soon. So I'm just going to run through a couple of the key elements of it here verbally, and I apologize. I know this is hard to follow along when it's just verbal, but really GMP is fairly simple. So I'll talk about the wash way in a second. So GMP is all about controlling the elements that impact the inputs to your end product by the consumer. So you need to have manufacturing processes that are clearly defined and controlled so that you've got consistency of your end product and you're in compliance with your approved specifications and procedures. So that's what GMP is all about. Those critical steps of the manufacturing process need to be validated so that you can prove over and over again that they are giving you the results that you're looking for. Your GMP process and certification has to include the following. So you've got those defined processes, you're validating them, you've got qualified and trained personnel, you've got adequate space, you've got suitable equipment, your your equipment and all of your handling materials are made of appropriate materials and products. You've got approved procedures and instructions, and you've got suitable storage and transport for that end product for the customer. So again, you don't need to memorize this. I'm going to have all of this in the presentation for you. Your instructions for your staff who are manufacturing your GMP certified product need to be very clear and easy to understand. And those operators need to be trained to carry out and document the procedures. So that's what GMP says you need to do. GMP is not prescriptive into how you do it. It says these are the results you need to achieve. How you choose to get there is up to you. Okay, so now I'm going to go into a little bit more about planning, but consider this in the context of what was just noted there on GMP. So there are a few different bullets on this slide here. So if you can get down to until it says floor finish, thank you. Okay, so the best advice I can give on the wash bay, if you are in the planning phase, whatever size you have allocated for your wash bay, triple it. I can promise you that you will not regret that decision down the road. And if you don't, I think you will. So most sites, what they do is they dedicate as much space as they possibly can to growing, because, okay, the more plant I grow, the more profitable we will be. Then they think, okay, well, I need to be able to process the stuff. So, okay, maybe we need to cut into the growing space for that. The afterthought is always washing. And what ends up happening is you're growing all of this material. Maybe you've planned enough space to process it, but the bottleneck always becomes the equipment that piles up in the wash bay. And what ends up happening is, without doubt, the number one spot where equipment gets damaged is in the wash bay. Equipment tends not to get damaged when it's in operation because your staff is trained to operate it correctly. But when equipment gets banged around in the wash bay, or there isn't enough space for it to dry, so people lean it up against things and then it falls over, or you've got clean equipment and you don't have a clean surface to put it on, so you're resting it on something else, that's where all the damage happens. That's where the bottleneck happens. So setting up your equipment, your wash bay so that it's large enough is the most critical thing. So you can consider these things here. So I talk about equipment size when broken down, how much space do you need, having an area for equipment to dry, access to hot water. If anybody on this call has not had an opportunity to try and clean cannabis resin off equipment or off gloves or anything else with cold water, try it. It doesn't work. You must have hot water. Rule of thumb for temperature, aim for no higher than 65 degrees Celsius. Most detergents are designed to function between, say, 55 and 65 degrees Celsius. If you go hotter than that, not only are the detergents not designed for that, but most equipment includes materials that would be impacted. So think adhesives and plastics and things like that. Even stainless equipment, you are going to have joints and things like that are going to be impacted. So try and max out at 65 Celsius. Your wash bay needs to have sufficient drainage. So we have a photo there of a trench drain. I find those tend to be quite good because they can carry a lot of water out of the room versus a single round drain hole. Air handling. I mentioned this at the beginning with respect to your processing room, but you need to have appropriate air handling. I was at a very large facility, around 1 million square feet, and they had not designed air handling into their wash bay. The people in there were using cleaning products, including their sanitizer was isopropyl alcohol, and you could see the cloud of ISO floating in the room. And it got to the point where they had to send some of their staff home with alcohol poisoning. So not planning in appropriate air handling in your processing space and your wash bay is a big problem. Floor finish, epoxy floors that you can easily clean is great. And as mentioned earlier, making sure that your equipment, your processing space, if you can, make it as close as possible to the wash bay. Okay, next slide. So there's a few different slides here, and I'll just tell you to click forward as we go through. So you want to have a number of racks and tables. If you can, get them with casters so that you can move them around and make them, place them where you need them when you need them, and get them out of the way when you don't need them. And think about it from this perspective. Again, if you are the one cleaning the equipment and you are trying to work on a piece of equipment and wash it on the floor, not only is it dirty, but it's very, very uncomfortable. You might focus on cleaning that equipment well for the first 10 minutes, but after that, your back gets tired and you begin to fatigue. So giving your staff ergonomically appropriate work surfaces is really, really critical. And it will ensure that your equipment actually gets clean and is not damaged. So that one's really, really critical. Okay, next one is a hot water pressure washer. Highly recommend this. Super helpful. Is the best thing for getting rid of cannabis resin. Next one, low pressure garden sprayer. These are helpful because you can douse your equipment. And I'm going to talk about surface contact time when we get to cleaners. And that's what this helps with. Compressed air, especially if you're dry trimming, hitting your equipment with compressed air first to do an initial clean off, and then coming in with your liquid cleaners is really efficient. Having drying racks for your equipment. Keep going with the slide. One more. And again. Okay, so specialized washing equipment. MOBIUS, we have a specialized washer for the tumbler and the brushes. For the trimmer, there are other pieces of specialized washing equipment. That's just an example. And then the next one is hand tools. So making sure your staff has plenty of supplies such as this on hand. Okay, next slide. So we're going to talk about cleaning a little bit here. And I think it's really important to start up front about defining what cleaning means. So a lot of people get these things mixed up. It's pretty simple. I pulled these definitions from the EPA, Environmental Protection Agency in the US, but you can look this up out of UDRA, you can look up Health Canada, whatever jurisdiction you're in. These are the industrial hygiene definitions of cleaning versus sanitizing versus disinfecting. So cleaning is removing the dirt from the surface of your piece of equipment, for example. Sanitizing is killing the bacteria on that surface. It's not intended to kill viruses. Disinfecting goes one step further, and it's killing both the viruses and the bacteria. So I'm going to give you a really, really clear example of why all three of these are necessary and why you don't just go straight to disinfecting. A number of sites that I've been to, their QC people are obsessed with disinfectants. No, back on the last page, please. So if you are disinfecting equipment and you're hitting it with ISO, you can do a swab test on there, and your swab test might come back clean for the bacteria that you're testing for or the virus that you're testing for, but you might still have cannabis resin on that piece of equipment. And I've seen this time and again where I can see the resin. The resin is clear enough for my eyes and everybody else's eyes to see, but somehow QC says that it's okay to go forward because the surface has been hit with isopropyl alcohol and it's been disinfected. Well, that makes no sense whatsoever. So back to GMP, you cannot introduce a contaminant into your end product. So if you're leaving cannabis resin on the surface, even though you've killed viruses and bacteria, that does not pass the test. So it always goes in this order. You clean first, you rinse, and then you sanitize, and only if necessary would you disinfect. Most cannabis regulations around the world, they're only requiring sanitizing. Okay, now we can go to the next slide. Okay, so when you're developing your SOPs, number of things here, and I realize that we're getting on with time here, so I'm just gonna let you guys kind of read these things for yourself. A number of different elements that you need to think about when you're writing up your SOPs. I've got a link on the bottom there to a Health Canada summary of what's required for developing an SOP. I've tried to source from a number of different jurisdictions for this presentation. So you'll see Canadian references, European references, US references. Every single jurisdiction has their own. What I can tell you, having read all of these regulations in great detail, is that the harmonization between them is very, very close. So use whatever you find is the most straightforward. Go back and verify against your own jurisdiction's requirements. But you can use this as a nice guideline to get yourself started, get things off the ground. Okay, next slide. This one here gives a very specific links for those 3 jurisdictions I just mentioned, starting with Europe for cleaning validation. And the cleaning validation is all about determining if your cleaning SOPs that you've developed actually work. So just because you've written something that you think works doesn't mean that it does. And that's back to that example I just gave you. You know, they were disinfecting the equipment, but they weren't actually cleaning the equipment. So if they went through a cleaning validation process, they would have been able to identify that gap, amend their SOPs to include some additional steps for that, come back, validate it, and make sure that it's working. So that's the key, and that's why cleaning validation is a part of GMP. Okay, next slide. There were some amendments I made to the presentation around here. So just think of this like your quality control program here. So your quality control program and people, you're going to be working with them to make sure that your cleaning program includes at least the following. So you have to have the written procedures for cleaning your equipment. You have to have a schedule for that. And you need to be able to prove it. So you need to be able to document the fact that you are doing this. I'm not sure if we have anybody from the US on the line today, or anybody's going to be listening to this. In the US, dry trimming is very common, not all over, but certainly on the West Coast. And when you're dry trimming, you don't get as much resin buildup on equipment. So what I find is that the cleaning schedule, when people are dry trimming, tends to be less frequent. And I think that that's a bit of a mistake because it's not only damaging to your equipment, but it's not a great habit to get into if you are going to move towards GMP. So if you're not doing a GMP certified end product right now, but you're thinking about it, developing a cleaning schedule that's got high frequency in it is would be my advice. Okay, so next up, selecting your cleaners. So the first one there is around selecting cleaners that are appropriate for the job and for the equipment that you're using. So you might be using different pieces of equipment and different materials for drying racks versus maybe your pre-roll cone holders or your trimmer. And so you're going to involve plastics, you're going to involve stainless, all those kind of things. So you need to have the right cleaner for the job. And then the second bullet there, I mentioned this earlier. So cleaners work based on surface contact time. So in order for a cleaner to penetrate a soil, it can't just be sprayed on and then immediately wiped off. The cleaner needs to be able to sit on there, and it needs to get underneath so that it can lift the soil off whatever the surface is. So your SOP should allow for that surface contact time of the cleaner to do its job. And then that last bullet there. So a lot of people think that more is better. And I can tell you that I remember my first year chemistry teacher. He was pretty fun because he believed in the more is better philosophy and blew out the ceiling tile in our lecture hall using his more is better philosophy. It was amusing, but not what you want to do with isopropyl alcohol. So 99% ISO is used to clean electronics specifically because it evaporates quickly. Okay, so that goes against the principle of surface contact time. So if you are using ISO as a sanitizer or a disinfectant on cannabis processing equipment, it's better to work with 30% or 70% ISO than 99% because it will not evaporate as quickly. So I recommend 30% ISO is more than sufficient for using when you're sanitizing cannabis equipment. Next slide. Okay, so I get this kind of, I actually just had this the other day from somebody who said that one of the regulators was asking them about this, and they were mistaken. So regulations do not dictate what cleaners you are allowed to use. The regulations simply state that you have to have a cleaner that is effective for its purpose and does not remain on, say, the processing equipment to be introduced into your end product. Okay, so there is no regulation out there for cannabis manufacturing, whether it's Europe or Canada or South Africa, wherever you are. They do not tell you which cleaners you can and cannot use. They just tell you that you need to use cleaners that are effective for the job and do not introduce contamination into your end product. So push back on that. Use what is appropriate for your equipment and for the material. Okay, I just want to skip over this slide because there's, go to the next one. Well, that I have specific links to GMP stuff later. On preventative maintenance, this is kind of the last thing here, and then we'll get wrapped up. The best way to manage your equipment is by being very proactive in how you manage it and not reactive. So rather than running your equipment into the ground, you want to take a preventative maintenance approach. And usually people think about preventative maintenance from the perspective of maybe changing out parts. For example, changing out bearings before they wear out, changing out belts before they wear out. And that's absolutely true. That's important. But on cannabis processing equipment, the number one thing that is going to run down your equipment more quickly than anything else is not cleaning it. So proper cleaning on a regular basis is the best preventative maintenance thing that you can do. Setting yourself up to perform that cleaning in an appropriate wash bay that's been set up correctly is going to be your number one partner in doing that. And then making sure you've got all the spare parts on hand and you're tracking your hours on the equipment so that you can be ahead of things is key. So an example on the MOBIUS trimmer, we rate parts based on the hours of operation. So we say, okay, this part is rated for approximately 200 hours. This part's rated for 1,000 hours. So as you approach that mark, you want to be looking at changing that out before the part fails and leads to other issues. I'll just pause right there for my hosts. That's kind of it on the content. The last one was just kind of an overview of the MOBIUS equipment lineup, but how are we doing on time, and do you want to maybe hit questions up first?
I think that we are run out of time. I don't have access now to chat to see the time. Okay. It's better to, regarding questions, because you are run out of time, we will send the questions from the customers directly to your email account, and we'll reply to them directly. So
okay,
in my opinion, just mention the product line of MOBIUS, and then we can wrap up.
Okay, sounds good. Well, if you can just go on to the next page there. What I will do on this presentation is it will be hyperlinks for the different machines that you see on the screen there. So the MOBIUS equipment lineup, we are focused on everything after the stalk has been chopped down. So the starting step after that would be bucking, so removing your leaves and flowers from the stalk, and then through into trimming and sorting the material. So you see those on the screen there, and we connect everything, automate everything with conveyors. And we also have a couple of different options there for milling product, and the mills are aimed at preparing your material for use in manufacturing extract products or pre-rolls, and they create a very, very consistent grind for those things. The flagship has always been the trimmer for us, so if anybody has questions on those, lots of information on the website, and I'll obviously have my contact information and that available for any technical questions. I'll leave it at that. Back in your hands.
Okay. So thank you very much, Amanda James. I will stop sharing my screen now because I think you have nothing else to show. Thank you very much for your presentations.
Thank you.
It was very useful, very helpful, and a lot of information in so less time. So I suppose next time we will need to have double of that time in order to be able to present all the solutions of MOBIUS. I will collect all the questions for you because I see in the chat room that there are some questions for you and some questions for CONVIRON also. I will collect all of them, and I will send you a mail in order to be able to reply to our customers.
Sounds good.
Perfect. So I think now it's time to wrap up and finish with the presentation. Also, Chris should appear on the screen also and say the last goodbye to the cat and thank our attendees.
Well, Hellamco's first Cannabis Excellence Day has come to an end. And I would really like to thank all of our partners for supporting this webinar, my colleagues, especially Yanis for his contribution. Yanis and his team will have now the difficult task, as he knows very well, to answer many questions and submit technically excellent codes. I must say your participation was overwhelming. We had around 360 people registered and more than 160 people attended live. In the next coming days, we will send the recorded version to all and the presentations also. So today we analyzed the many aspects of this complex plant and the correct way to cultivate, dry, trim, and test the quality or do research. All solutions that you saw are not theoretical, but as exhibited, are in successful operation in major cannabis facilities all over the world and also in our market. Our experienced scientific and technical team remains at your disposal for any additional info or analytical quotation for equipment to support your goal for the highest quality products. Of course, the global situation with logistic problems and price increases of raw materials really tests manufacturers' capabilities to deliver within our customers' time frame. Therefore, decisions must be taken as soon as possible, as first come, first served rule applies without exemption. We cannot change this. We mostly overcome these challenges, and we are optimistic as we see an expansion in the field of pharmaceutical cannabis in Greece and North Macedonia. So we wish you all the best for your future plans in this cannabis industry and wish you all global peace and prosperity. Thank you very much.
Thank you very much also.